One was amazing. Day 2 is going to be potentially even better. Don't know how we top day 1, but we're going to try. So just repeating our logistical things in case you weren't here yesterday. There are bathrooms around the corner on the first floor. If you're on the second floor, there is a key code to the bathrooms that are kind of in the same place. And the key code is on the board in the poster room. Poster room is up the stairs to the left. No food or drink in the auditorium, please. If you have water, that is fine. So I'm going to assume that any beverage in a travel mug is water. Don't prove me wrong. So yes, we're very excited to be here again. The meeting is being recorded. So if you are asking any questions during the meeting or participating online, asking questions, there is the chance that you will be caught on camera, so just be prepared and don't say anything that you do not want preserved for posterity. The Wi-Fi code is CNRA guest, capital CNRAG, rest lowercase. We have coffee and tea outside, sponsored by ICF. Huge thank you to ICF for paying for that, given the budget crunch and rules about buying coffee when you work for the state. We need an outside sponsor for that, and we're very grateful. But please do enjoy that coffee in the lobby and not in the auditorium. If you need any kind of accommodation, help getting upstairs, you do need a key card to use the elevators. Just talk to Christine in the lobby at registration. Huge thank you to all the volunteers who helped put this together. We have mic runners and timekeepers every day. We have folks from the California Natural Resources Volunteer Program assisting with registration so that none of us have to miss part of the talks. And Denise Goodman has coordinated all the volunteers, so huge thanks to her. Huge thanks to all of the rest of the organizing committee for the workshop. We put together a great program and couldn't have done it without you. Especially huge thanks to Christine Job and the rest of the IEP support team who really did all the hard work so that it was very easy to just put the program together. And I realized yesterday I didn't introduce myself. I'm Rosemary Hartman, and I was the program organizing chair for this year's workshop. And I'm the IEP synthesis lead. So talk to me anytime about data integration, data, zooplankton, where I got my dress, etc. So we're about to start our plenary talk for the day, but I want to just give one last little plug. Right before today's poster session, we're going to have a poetry slam again. We didn't get one last year, but we're trying to reboot it this year. So if you would like to pitch your poster with either a just brief prose description of what's in your poster and why people should visit it, a brief haiku, limerick, poem, skit, song, etc. We'll just have people line up before that break in a row and cycle through all of the poster pitches pretty quick. So stay tuned. All right, then. Without further ado, I'd love to introduce our plenary speaker for the day, Felicia Marcus, who comes to us from Stanford's Water in the West program. And she was previously on the California State Water Resources Control Board and has a long history of using science for real water management topics. So she's here to talk to us about how we can make our science more management relevant and get it in the hands of the people who are making decisions for water in California. And then if we can switch over the presentation to Felicia and let her take it away. She really wished to be here in person today, but had a last minute change of plan. So we'll be presenting virtually.
There we go. Hi all. I'm sorry I can't switch to my picture, so you can't see me in the in the corner a little bit, but really, really happy to be here. And I want to thank Rosemary for inviting me, and Steve for his help too. I really wish I could have been there in person, but I had a work conflict for travel I had to do for an energy board that I'm on that I couldn't miss. The abstracts look great. I skimmed through them and I actually understood some of them, many of them, thanks to the training of many of you in the room that helped me do that. And also those of you who wrote them in a way that someone without a scientific background, but who cares about science, could understand. And that's a bit of what I'm going to talk about today. You know, your work is really important, and it kind of goes without saying. I just want to say it as a former policy maker at the state, federal, and local level, where I've been lucky enough to be able to work on issues that involve the integration of science and policy. But I've also been lucky enough to have tutors, mentors, and guides from the scientific community who happen to be really good translators with a lot of patience for folks who did not have scientific training coming into it. So to the extent that I give any meaningful advice or tips to you, know that there are a lot of other people, whether it's Bruce Herbold or Aaron Forsman or any number of you that are there, who have just shown me a tremendous ability to be able to translate between science and policymakers. But I wanted to also start by saying, I know there are times when you must feel unheard and unrespected and unappreciated as policymakers who get to make decisions, frequently don't fully understand the science you're talking about it, sometimes don't care about the science, sometimes twist the science. And I just want to say you are not alone in that. You have a lot of friends, and that it's not just between scientists and policymakers. A lot of times between policymakers and politicians, it's between any two sets of players that have a focused look. So I really just want to start by saying that I really do appreciate and respect the amazing work that you do, and that I think it becomes even more more precious and more important as we move into the coming era, whatever we're going to end up calling it. It'll be something beyond the Anthropocene because it'll have a harder edge to it. And I also hope I can just give you some insights and tips to follow on Steve's really excellent presentation yesterday, which I got a chance to watch, and I was scribbling furiously. So with no further ado, here's what I'm going to talk about a little bit about me, but not a lot, because I know a lot of you, although I've been gone 5 years, so I don't know all of you, and I never knew all of you, but I'm sure I know a lot of you in the room. What's at stake, a reminder about the challenge of connection and bridging for those of you who've been at Delta Science conferences and other conferences over the years where I've been asked to talk about this topic. You'll remember some of these stories. I have some renewed tips on how to do it, with an emphasis on how scientists can better communicate with policymakers. When I'm talking to policymakers, I give them a similar but different set of tips, and I spend a lot of time on this, sometimes doing an hour-long training. So I'm going to go pretty quickly through it just as tidbits and thoughts, and I can share the slide. I'm always happy to follow up, as people know. And if there's time, I'll give you a few story examples. Some of these things may seem basic, but I will tell you that over 30 years of working on projects, they bear repeating for many people, including myself. I won't go through my whole career. A lot of you know more about it than you ever wanted to know. I started out as a public interest lawyer and organizer in LA, private sector lawyer as well, doing patent cases, where I first learned how to translate between engineers and others, similarly in the city of LA, and I'll talk about that a little bit. Had the good fortune, some thought misfortune, but the good fortune to be in appointed positions in government and public works in LA, probably the hardest job I'll ever have. EPA during the Bay Delta Accord years of the 90s, the state board during the Brown administration, some time in national nonprofits. And now the very purpose of my job is to bring a practitioner's view into the academy. And certainly being in a research university with lots of scientists and engineers and geophysicists, etc., I'm getting a full immersion into that world and different methods of presentation, different methods of communication, and it just has reinforced some of my views and given me others. The Ninja Turtle is there for a reason. Obviously, they live in the sewers and eat pizza, but that's me in that suit. During the Santa Monica Bay Restoration Project, which was a really good project, has a new name now. In the 80s, when we pulled it together, and this is a science thing, probably the legislation I'm most proud of it was when we got Santa Monica Bay into the National Estuary Project. As you all know, and most people don't realize, it is not an estuary, but it is a legislative estuary. And the reason we did it was to force a table where the policymakers, the activists, the scientists, people from different departments would have to come around a table and be able to talk about what to do about Santa Monica Bay. So this has been important to me for, quite a long time. Other bias that people know, in theory, there's no difference between theory and practice. In practice, there is. A lot of people are mad about this, particularly very smart people in academia, even more so than in folks who are in agencies and doing science in the public domain. I found railing against the heavens about how we do legislation or policy regulation. It's just the way it is. You've got to get the votes. You've got to get something done. It's all about people. So you can either rail against the heavens, or you can figure out how to work with it to have impact. And many of us have had to learn how to work with it. The need, climate change is just going to get worse. We're going to need to adapt faster and more thoughtfully. We have both the Sierras, where we see the pictures all the time, but also the Colorado coming to the end of its train. I could talk about that the whole time, but you all know this more than the average bears. And then, of course, our ecosystems plummeting. So I could really just say, listen to Steve. I thought it was great. And I've just highlighted some of the ones that really dovetail with what I wanted to talk to you about. And part of it is this constant dialogue is fundamental. It's not one perfect presentation. It's a conversation, an ongoing conversation, getting to understand each other's needs and skills and having mutual respect. That's not that easy. I'm not saying, ask a policymaker out for a beer every other day, but find those opportunities to come in and just talk. What are they working on? What do they care about? What's important? What do they struggle with? Ask them questions so they get to see you as a person, as opposed to feeling sometimes like, especially very smart, very accomplished engineers, scientists, you can insert other titles, are talking at them, or worse yet, talking down to them. Folks in policy jobs are sitting at a dais and seem to have power, but they're just as insecure as everybody else, particularly around really smart people like you. Sometimes they don't care, but a lot of times they're just intimidated and they don't understand. And people tend to hear a negative and a condescending negative, particularly if you don't smile. I'm not saying smile in that forced sort of a way, but you do tend to have more introverts in the sciences and engineering. I'm going to make a gross generalization. I'm going to talk about this in a moment, but it is important to avoid what a colleague of mine at EPA used to call the Easter Island effect, where a policymaker is talking with you and you have just a blank expression on your face. Now, I know from lots of Myers-Briggs training that there's a world of things happening behind that sort of lack of expression, and that people can be listening better than the people that are nodding and smiling. But you do need somebody to nod, smile, to ask a question, to know that they're listening, because a lot of policymakers feel they're being blown off. I mean, there's a lot to this, but a few things to be really aware of. And the science matters less than we think is just that politics, policy, and other factors come into play, and you just need to be aware of that. You don't have to like it, but you need to be aware of it. But if the science isn't intelligible to a non-scientist, then it's harder for them to integrate it into their work and their thinking. Many of you have heard me talk about this. There's a great Chinese expression. I won't torture you with my ancient Chinese from college, but it's a phrase like a chicken talking to a duck, that people can seem like they're speaking the same language, but they're not speaking the same language. And you've heard me say over and over again that the challenge isn't as much ecosystem management as it is ecosystem management. That just means paying attention to the people that you're trying to influence or that you're engaged with across a table. And it may seem obvious to many of you, but after 30, 40 years of doing it, it happens every day where you see people not actually getting it and getting frustrated. So 2 of 10 examples I could give you here is, for example, that people are speaking a different language, even if it's in English, and that people are likely to hear what they expect to hear rather than what you're saying. Just 2 things. And the point is you have to think about it. And the key thing for you all, being the smartest guy in the room is not helpful all the time, particularly if you convey that you think you're the smartest guy in the room, nor is being the loudest or angriest person in the room. This is, the angry guy is more for my colleagues in the environmental world, which I come out of, that don't realize that's not the most effective way to win friends and influence people. Both of these might feel good, but they're not really helpful. Some of you have also heard my 10 dysfunctions of smart people talk that I developed when I was at one of my jobs, where I realized that unless I framed how to be more effective in terms of smart people having this affliction, that smart people wouldn't listen. And it worked actually like a charm. Things like all you know is all there is, knowing that nobody likes the smartest guy in the room. And if you think you're the smartest guy in the room, you probably aren't. It's like mastering level 3 on a video game and thinking there are only 3 levels. There are always more levels. There's always someone who's going to be smarter than you in something or other. It may even be your field, and you won't even know it. And so the smartest people are always looking for smarter people in the room and come at it with humility. Other things some of you have taught me on this is the average person doesn't think in exceedance plots. These tell a whole story to a lot of folks at the state board. I can follow them now, but people were surprised how long it took me to get them. Now, I may be significantly challenged on graphs compared to other people, but I'm not dumb. And I know a lot of stuff, but you have to explain the chart. You have to explain the axes and what it means. You may wonder about the dolphin. Obviously, this isn't really a chart about dolphins. It's a chart about how to draw a dolphin. But some of you have heard my story where one of the staff at the water board was retiring, and I said, oh my God, who's going to explain the charts to me? And he was so good at it. And he told me a story about how he was gone and someone came up to brief me. And afterwards, when he came back, I mean, he just couldn't make the meeting. He asked the person, how the briefing went. And the person just sort of put his fists on his hips and said, she said the chart looked like a dolphin, because that's all I could see in this chart without it being explained. So you do need to explain it. And then the Queen Mary stories, obviously, where at the San Monica Bay Project, we argued, we actually went on this boat with the scientists and the policymakers and we must have spent 2 days, I think it was 2 days, and by the arguing about how to write this report. And by the second day, I finally said, let's just write it both ways, because we're trying to write a scientific paper and we're also trying to influence policymakers. And that's what we did. Simple solution, took us 2 days to get to. Other examples that some of you have heard, this speaking a different language, when I was a lawyer dealing with the city, it's a much longer story than this. A judge forced the City of LA people and Heal the Bay, another activist, into a room. Essentially, we had to meet for 3 days in a row in a fight over the treatment plant. And it meant that the city folks had to answer all our questions, but we had to listen to the answers. And I felt in a meeting that the city engineer, who's a very well-regarded engineer nationally, was blowing us off. We didn't understand his answers. He was being condescending, et cetera. And I felt like a brat as a lawyer. And I said, or just as me, and I said, I'm just going to keep asking this question a different way till he answers it. And I finally asked it like 5 times. I was getting more and more irritated, but I was going to keep doing it. finally asked it one way and he just answered the question. And I realized, oh my God, he was answering the question, but in engineering ease. And that was a key breakthrough in the relationship between Heal the Bay. And a lot came from that. Similarly, at Lopez Canyon, which was a landfill, we'd done pretty well with the community, but then I heard that things had sort of fallen apart. I went up there and just to watch the meeting, and sure enough, someone asked a question about what kind of flood the 100-year flood would look like, and the engineer doing the speaking answered in terms of cubic feet per second flow, and you could feel the irritation there. My God, okay, I know what the problem is here. And then his boss stepped up and said, well, I can explain it. And he thought for a minute. I thought, oh, great, this is going to solve it. And he said, yeah, it's, X amount of cubic feet per second flow. And I finally went, okay, this is the problem. She wants to know, is it like the flood of 85? Is it like the flood of 72? And then I explained the square basketball thing of cubic feet per second. But again, that's not a language that regular people understand. Another thing is that doing another model run is not an answer to a policymaker. It is to a scientist who knows how to model and to a modeler, but just saying an answer to a question, you'll do another model run, I have seen it enrage state board members who feel they're being blown off, when in fact, the modeler was really answering the question in the way that was his language. And he was a wonderful person. And then I had to have a separate meeting and explain what she was asking. And part of what you do there is you ask a question rather than just give an answer. And then I know I'm running out of time. People do hear what they expect to hear. So you need to try multiple ways to answer a question, and you need to ask questions to understand it. The smog check story I've told a number of times, where it was all out war in the 90s with the state of California and EPA. And I went personally one-on-one to meet with lots of legislators. And one in particular told me EPA was trying to do something really outrageous. And I said, no, actually, I know you're being told that, but we're not. We can be more flexible. I'm making a long story short. I went through a dance with him about 3 times where he thought, and he goes, no, EPA is requiring. And finally, I pulled out my EPA name tag and showed it to him with my picture and my name on it. I said, I swear to God, I work at EPA. We're not. And that was the breakthrough in the smog check negotiations. And it took a good 15 minutes. So you can't assume that people have heard what you've tried to say. In Bay Delta, you've all been there. It's so complex, and the talking point wars are so complex that some of the message, and this is my particular beef, and I know many of yours, about the importance of flow, just gets swamped. Now, it's true there are multiple stressors, and many of you have done great work for decades on this. And PPIC even did a great review of stressors, showing that scientists in general thought flow was the master variable and most important. That has not popped through in the airwaves in a way it needs to for us to move to a resolution, whether it's voluntary agreement or just regulation, that has flow included along with other activities that can hopefully help fish faster. I'm totally oversimplifying it because we're out of time, but just to give you an idea, here's some tips, and this may seem obvious for you, that I want to leave you with. Think about mindset management. There's a great book on it that even teaches you questions you could ask somebody that you want to influence. As I said, not just scientists and policymakers, but you have to be mindful of yourself. What are your biases? What's your language? What's your framing? Try to learn the others by asking questions. Myers-Briggs, which many people in the scientific and engineering community like, I think because it has formulas and matrices, is incredibly helpful and has been a lifesaver to me over the years. Covey, also good, trying to understand others first before being understood and repeating yourselves louder and slower, really important for everyone. But again, you have to learn enough about the other person's language to not misunderstand their intent. And again, as I said before, all you know isn't all there is. Be as curious about the people as the science. People have to approach each other with respect for their different skills and roles. And I always say, don't dismiss the power of story. It's powerful. It's the only thing that changes things in the world. Integrating science into it is good. But I also tell others not to dismiss the power of science well communicated to an audience. And again, as I said, do documents 2 ways or longer executive summaries, FAQs on science documents. Look at IPCC, which is a mass of science, always does a summary for policy. makers, and of course, frequent conversations. Again, look at graphs and charts from the non-scientist vantage point. Be empathetic to those of us that don't just know that. Be willing to describe what each axis is and what you see in the chart. Don't assume it tells the story. Things like particle tracking videos, bubble charts, etc., are much easier for those of us who don't have a scientific background. Have empathy for policymakers. It's a hard job. We're wedged between the politicians and the public and everyone else. I know sometimes we don't have enough time. It was great to be able to spend days at meetings like this in my job, but even then I didn't do it as much as I would have liked to. Bring some humility rather than disdain for policy. And I do the same thing back the other way. Really important when you're talking, try if you can to start with some key conclusions and spend less time on your process and methods. I know that's really important in the papers that you share with each other and in the academic world. And you need to be ready to explain all of that or put it in appendices, but that's not the thing you're trying to communicate to a policymaker. And even if you can't reach a conclusion, which you rarely can, try to give a sense of where the science is going, or something. Options are a good way to go. Options, pros and cons, I still owe Dick Norgard a cup of coffee for being rude to him at a Delta Council meeting where I was frustrated there were no conclusions. But it's really important to a policy. They just want to know what to do or what's more likely than not. Again, try also not to have the only conclusion be more science needed. That's frustrating. That gets to the point that Steve talked about it costs a lot of money. This can be frustrating for policymakers. Stop for questions periodically. Ask a question every now and then. Remember, people need to be heard and understood. And sometimes you may misunderstand what they're saying too. That's a whole other story. And ask yourself, what's the story? I lost 2 slides, sorry. The folks who've done work on beavers have done an extraordinary job making the science clear and fun and usable, and you've seen an amazing transformation. I did a talk myself just Friday at Stanford to a bunch of very technical folks, and a couple of the slides about a lot of things, but about beavers, and I forgot to explain why beavers make their dams, and people came up afterwards to explain why they make I hadn't done that basic picture because I thought it was self-evident, had spent so much time on beavers. Dr. Emily Fairfax, now at, I think, University of Michigan, not CSU Channel Island, does this whole Smokey the Beaver thing that has made this stuff accessible. Somewhere, and I don't know what exactly, could be Chuck, could be childhood, could be a tribe, the governor understood the importance of beavers, and there's been a total transformation in it. He didn't understand all the science, though I'm sure he does, but an amazing turnaround on a really important issue for meadow restoration. One final example that I think is really good are the true confessions. My husband works at SFEI. He's as much a fan of this work. He didn't do it himself as I am, but they did a great job trying to make the case for using terraced wetlands and fill to deal with sea level rise, to buffer that force rather than seawalls. And they used a metric of how many Empire State Buildings it would take to get enough fill in, which people can relate to if they're policymakers. Similarly, a couple of the staff there that get a gold medal on this stuff, Julie Beagle and Laura Tam, who was then at Spur, and is now at the Resource Legacy Fund, get a real gold star because they were being pushed to get this adaptation atlas out quickly, and they refused for 2 reasons. One, as you can see, this wasn't why they refused. Really good graphics that give you a sense of the possibility. It's filled with science, but a sense of the possibility, and they broke down every area of the bay to show people what they could do, what the art of the possible was. But what they did was they refused to put it out until they went and had conversations and met with all of the policymakers who were going to have to use it. So by the time they rolled it out, it was not seen as an SFEI Spur report. It was seen as the collective communities report. Just a great example of doing this work well. And with that, thank you. Sorry, I talked quickly because I had to get through a lot. But I hope that some of that is helpful, and I'm very happy to follow up, and bless you for the incredible work you do. And it is my fervent hope that you get listened to far more and far more often, because we need you to deal with the complexity of adapting to climate change, let alone fixing the issues we know are right in front of our face today.
Thank you so much, Felicia. Some really inspiring and thought-provoking words. We have a few minutes for questions. Does anyone have any questions for
great.
Felicia?
I can't unshare it, unfortunately, and I can't turn on my video. I don't know what I need to do to be able to do that. Have you gone back to me?
You're still live in the audience, but we now just have the workshop logo on the screen.
Okay, fantastic. Okay.
Any
just
questions for
listen.
Felicia?
Could you tell the
Bruce.
Chinese story, please?
Bruce would like you to tell the Chinese story, please.
Oh my gosh, thank you. No, that was the first one. I was thinking, I was talking, I left it out. It's just the where I first, this is even well, I don't know if it was before the Bob Horry story in LA, but engineer, but no, I think it, yeah, I think it was for the smog check story. I was in China, I was a Chinese studies major in college, and then my Chinese was far better than it is now, although I've gone a few times and I can sound good, but I don't, it's called tingbudong. I can hear, but I can't understand it quickly. So I was on a relatively long trip in China. I was waiting for a plane in Chengdu to go into Tibet. And there was another woman who was stuck there for a long time who had lived in China for 15 years, non-Chinese American, clearly not Chinese. And her Chinese was gorgeous. It was really good. So I said, will you listen to my Chinese? I'm having this problem where I'll talk to people in Chinese and some will understand me and some won't understand me at all. And she said, oh, I know what's happening. I don't have to listen to you. And I said, well, what's happening? She said, well, they're looking at you again. This is a long time ago now. A lot of everybody, it seems like everybody speaks English in China now, but then not many people did. And they're taking one look at you and they figure you can't speak Chinese, so they can't hear the Chinese. I said, well, what am I supposed to do about that? And she said, you get right in front of their face and you say, slowly, I am speaking Chinese now, which seems ridiculous, but I do it. And all of a sudden people would throw up their hands and start talking Chinese to me really fast and tell me how good my Chinese was. And so it made me realize you have to do something when you sense people can't hear you. In the smock check story, that's why I held up my ID tag. I remembered the China story. He knew Felicia. He believed Felicia because I'd been in there so much, but he didn't believe that the great evil demon EPA wouldn't do what he thought of as a really stupid thing. But once he realized it was me and that I was at EPA, it created a breakthrough. So figuring out how to do that's really important. And those of you who know me know I always talk about coffee shops and bars because you got to take people, when you're a manager, you have to do it too. You have to take people one-on-one. You don't always have to buy them food or drink, but you need to sit down in a place and ask them questions and get to know them so they can see you as a person, and then they can actually hear you. Otherwise, they can't.
Thank you. That's really interesting, thought-provoking way of approaching things. Any other questions for Felicia? All right. Well, thank you so much for joining us. Really appreciated all the stories and a lot of different ways to think about relating to managers, relating to each other, and relating to people in airports.
There you go. People are people.
Yeah, exactly. Exactly. Thank you so much.
Thank you.
Yeah. Sorry. Hope you have a lovely rest of your day, and sorry you can't be in here in person. Okay, so now switching gears, we are going to have our first scientific session of the morning on Delta Smelt experimental release. And our session chair to introduce the first speaker is Denise Goodman.
Uh-oh. Peter wants to restart. Welcome to the Smelt Supplementation session, and we're going to jump right into it with Katie Osborn with Department of Water Resources.
Thank you, Denise. And thank you everyone for coming in person or virtually. I'm Katie Osborn with Department of Water Resources, and as Denise said, I'll be speaking about the first 3 years of experimental release in brief with some initial results from year 3. Before I begin, I'd like to thank my co-authors, Brian Schreier and Daphne Gill, and Rosemary Hartman, whose name should also appear up here because of her invaluable assistance with the analyses. And I also want to just acknowledge what an incredible collaborative effort this is. The people who have contributed are truly innumerable. And together, Department of Water Resources, California Department of Fish and Wildlife, UC Davis Fish Conservation Physiology Lab, U.S. Fish and Wildlife Service, the Bureau of Reclamation, and the Geological Survey have released over 190,000 cultured delta smelt into the wild since late 2021. So, oh, does the down button not work? How do I advance? Oh. Okay. So just a brief overview of years 1 and 2, and in the upper right corner, you'll see Y1, Y2, and that'll appear on any slide pertaining to more than 1 year of data. So year 1 was our first time releasing delta smelt into the wild, cultured delta smelt. So it was all about learning. There were over 55,000 delta smelt released, starting with a soft release event at Rio Vista, followed by 8 hard release events at Rio Vista, Suisun Marsh, and Sacramento Deepwater Ship Channel. Year 2, which was water year 2023, over 43,000 fish were released, and it was all about improving on those methods piloted in year 1. So each of the release groups was individually and uniquely marked to that release group using VIE tags and adipose fin clips, and UC Davis made improvements to their soft release enclosure design, and that was deployed as well. So in year 1 and most of the releases in year 2, fish were transported in carboys that each hold about 200 delta smelt. This is a pretty obvious limitation. So for year 3, one of our objectives was to pilot the large-scale transport of delta smelt. We planned and executed 2 truck releases using hatchery tanker trucks from Coleman National Fish Hatchery, and this was increased by 2 trailer releases for a total of over 50,000 fish, hard released via large-scale transport. Our other objective was to compare hard and soft release methods. We had 4 paired events planned. 3 were successfully executed. And for these events, all of the fish were transported in those carboys I previously mentioned. And the grand total of fish released this year was over 91,000 fish, so well above our production goal of 75,000. So a little more detail on those objectives. Large-scale transport pertains to both the trailer on the left, that's a photo from year 2, the DWR fish haul trailers, and hatchery tanker trucks on the right. This is the larger of the 2 trucks from Coleman National Fish Hatchery that we used for large-scale releases this year. And our other objective was to compare hard and soft release. So maybe I should define those terms. So hard release, as we see with the black bear, is when an animal is released from captivity directly into the wild without any sort of acclimation period. This is what we're used to in fisheries. Hatcheries typically release fish directly into the water, and it's a really cost-effective way of getting animals into the wild quickly and easily. In contrast, soft release includes an acclimation period in a predator-free setting so that that captive individual can get used to their new wild environment. So here we see Mr. Pine Marten is debating whether he's ready to exit his nice little shelter. These more intensive logistics obviously have increased labor requirements and costs, but soft release is highly favored in conservation biology because numerous wildlife studies have shown benefits to soft release, such as increased fidelity to the release site and more adaptive behaviors. So for Delta Smelt, our soft releases include an underwater enclosure that is installed ahead of the release as shown. Once it's installed, we come out with those carboys previously mentioned and shown there, load it up with fish and put a lid on it and let them acclimate for up to 48 hours. After the acclimation period, the cages are dropped and the fish are given time to swim away. Once it appears from the ARIS photography that the fish have mostly cleared the area, the cages are refloated. Hard release proceeds in much the same manner, except instead of using those carboys to load the cages, the fish are simply released directly and gently into the water. So to reduce sources of variability, we did all of our releases at Rio Vista this year, and we had 10 release groups, each of which was uniquely identifiable with VIU tags or adipose fin clip. And now into data. And before I get too into the data, I just want to caution against drawing any trends from the data that I'm going to show today. Our recapture rates are really low, and we simply don't have the statistical power to draw those trends yet. Okay. Warnings out of the way. On the top, we have released fish represented by some individuals released during our second trailer release this year. And on the bottom, we have recaptured fish represented by an individual caught in the Chips Island trawl. And in water year 2023 on the left and water year 2024 on the right, we see that we recaptured fish that were released using all of the release methods implemented in that year. So that's nice. And that fish were recaptured in similar proportions to those in which they were released. Looking at water year 2024 in particular, we see that we caught slightly fewer of those truck release fish than we might have expected. And we caught slightly more of those paired hard and soft release fish than we might have expected. And looking at trailer releases in particular, we see that they were 14% of the fish released and 14% of the recaptures, except that all of our recaptures come from the second of those trailer release events. And this just points to the high variability of our data and the importance of those environmental conditions at the time of release in influencing our recapture rates. So looking at fish distribution, I told you it was a low recapture rate. We've had 49 recaptures out of that 91,000 fish. And they're widely distributed throughout the Delta, mostly in the North Delta Arc, but we have had some fish exit the North Delta Arc and end up at federal salvage facilities. And this is similar to last year, and also similar to last year. This year was an above normal water year. Last year was a wet water year. So under those favorable conditions, we would expect this kind of wide distribution. We also had 2 fish caught in EDSM that were genetically confirmed as wild, and they were successfully transferred to the FCCL for brood stock. Speaking of EDSM, they have switched to their larval and juvenile gear, so we are unlikely to get more adult recaptures, but it's not impossible. We did get a recapture on June 28th of last year of a fish that had been in the Delta for over 6 months. So So one of the questions that we're interested in with releases is site fidelity. And here, please note that large scale in the middle pools both trailer releases and truck releases. So we would like to be able to target our releases to favorable habitat and have them remain in that North Delta arc where there's low risk of entrainment. And soft release is hypothesized to promote site fidelity amongst released organisms. And here there's really wide confidence intervals. It's not statistically significant. And note those outliers, but we do have a tighter grouping for those soft release fish, which is in line with what we would expect to see. We're also interested in distance between recaptures. We know that Delta smelt are a shoaling species. They do better together. And soft release is also supposed to promote adaptive behaviors, such as grouping in a shoaling fish species. So again, the grouping is not as clear as it was in the last graph, and it is not at all statistically significant, but we do see a slightly tighter grouping in the soft release fish, which is in line with what we would expect to see. And finally, I mentioned the importance of environmental conditions earlier. So here we have the recapture rate for the release on the y-axis and the turbidity for the week following the release on the x-axis. And when conditions are more turbid in that week following release, we tend to have a higher recapture rate for that release event. It's pretty wide confidence intervals, so we're not sure what exactly the shape of that relationship is. So we're going to be continuing to track this and asking questions about how environmental conditions impact our recapture rates and our release success. So in closing, this has been an incredible collaborative effort. There's truly too many people to name who have contributed wonderfully to experimental release. All release methods have led to recaptured fish, but not all release events, which points to the importance of the environmental conditions at the time of release. Overall, we've had a lot of successes. We've gotten a lot of fish into the Delta. We're seeing them move around the Delta. We're seeing them survive, in some cases, months in the Delta. But continued low recapture rates means that we don't yet have the statistical power to say which release methods are most beneficial to Delta smelt. And this brings me to our final goal, which is to optimize that cost-benefit ratio. So this year, large-scale transport was highly effective in releasing large numbers of Delta smelt quickly and simply into the Delta. And soft release is hypothesized to promote site fidelity and adaptive behaviors. So we're looking to balance those two things, the increased costs of a more intensive method like soft release with large scale. So what's next? Well, what's going to continue is uniquely marking each of those release groups, and so that we can learn which release groups are faring better and start to ask questions as to why that is. What you're going to see more of, we're going to be doing more of those large scale releases. We're going to be increasing production as facilities allow and continuing to look into environmental conditions. And what we're looking forward to in the future, right now, all of our tools depend on recaptures. These data are readily available. They require minimal post-processing, but we're developing other tools such as acoustic tagging to look at distribution and movement behavior, and genetic tools so that we can look at how well these hatchery releases are contributing to the next generation. So finally, I just want to close with a huge shout out to everyone who helped, whether it was in the field, lab, office, or all of the above. It is greatly appreciated. And I'll leave you with a little video of Delta smelt. Apparently, we have time for one question.
Very short but logistical question. Are you ad clipping every fish that you release?
So the ad clip group, that's the quickest marking method that we use. So we used it on our largest release group. So that large tanker truck that you saw, that was used for a release of over 25,000 fish, and all of them were ad clipped. The rest of the fish are, they are all VIE tagged, which is a liquid polymer that's injected in under the skin.
Uh, Thank you, Katie. And next we have Nick Hudson with UC Davis.
Hi, everybody. Thank you so much for coming. Today, I'm going to be telling you about quantifying the stress response of delta smelt to inform population supplementation. My name is Nick Hudson. I'm a master's student at UC Davis in the fungi lab. So as many of you know, delta smelt are endemic to the Sacramento-San Joaquin River Delta. And delta smelt were once abundant throughout the entire delta, but due to a variety of anthropogenic disturbances to the delta environment, delta smelt are now on the brink of extinction. In response to these population declines, a refugial delta smelt population has been maintained at the UC Davis Fish Conservation and Culture Laboratory since around 2007. And as Katie just told us all about, recently production at FCCL has accelerated to support a population supplementation program where these cultured fish are being released to the wild. And since 2021, almost 200,000 fish have been released. So I know you just learned a little bit about this, but I'm going to explain it again anyway. So supplementation looks like this. Initially, there's a period of handling when delta smelt are removed from their rearing tanks at FCCL and placed into these sealed transport containers called carboys. These carboys are then transported out to release locations for around 2 hours, first on trucks and then on boats. And then carboys are released to the delta using one of two methods. The first is hard release, where these carboys are emptied right out into the open water and the fish are allowed to freely swim away. And the second is soft release, as we just heard, where instead these carboys are emptied into acclimation enclosures. And there's also a third release method that Katie talked about, those trailer releases. So my study focused on this soft release methodology, so I just wanted to explain it a little bit more. So delta smelt are placed into these enclosures, and enclosures allow for the passage of wild food items. Delta smelt experience high survival and also positive growth inside these enclosures. Obviously, they exclude predators from reaching the delta smelt, and we also think that they may encourage natural schooling behavior and also site fidelity. So across supplementation, we see these distinct changes in water chemistry. So delta smelt are reared at about 0.3 parts per thousand of salinity. And then when they're placed into carboys, these carboys are salted up to 5 parts per thousand. And the carboys are also supersaturated with oxygen. Across transport, we know that within this carboy water, pH goes significantly down, dissolved oxygen goes significantly down, alkalinity and temperature stay about the same. And what this leads to is a massive increase in PCO2, or the amount of dissolved CO2 in the transport water. So essentially across transport, these carboys are becoming acidic. Upon release, water chemistry conditions return to normal, pH goes up, dissolved oxygen goes up, and PCO2 falls back down to normal levels. So we think that across supplementation, delta smelt are being hit by 3 distinct stressors. The first is handling, when they're being physically removed from their rearing tanks and placed into carboys. The second is this transportation process, as these delta smelt are being jostled around as they're moved from the hatchery out to the release locations. And then the third is these shifts in water chemistry. First, that massive increase in PCO2 during transport, and then the decrease in PCO2 when they're released. And we think that these three stressors are going to lead to a stress response in delta smelt. But what is the stress response? So generally, the stress response is known as a suite of physiological mechanisms to perceive stressors that threaten an animal's homeostasis. And it's primarily a process of rapid energy mobilization to allow that animal to confront a stressor and regain its homeostasis. And mechanistically, what this looks like is initially there's a period of rapid cortisol circulation that signals the rapid production of glucose. That glucose is then metabolized very quickly as a source of energy, and then lactate or lactic acid is built up as a byproduct. In responses to changing water chemistry that might stress a fish out, they have these specialized cells on the surface of their gills called gill ionocytes that remove or absorb ions to maintain the acid-base balance in the blood of the fish. And generally, these stress response mechanisms are known as adaptive and beneficial responses that are necessary for fish to live in stochastic environments. However, if a stress response is overly intense or prolonged, it can start to deplete the energy stores that a fish has. And ultimately, this leads to limited energy for nominal functions and adaptive behaviors and can threaten the survival of a fish. So therefore, we know that the magnitude and the duration are the two things that really mediate the detrimental effects of stress on fish. So going back to the stressors that are hitting delta smelt during transport, we think that handling and transport together are going to elicit a stress response in the form of increased cortisol, glucose, and lactate. And these changes in water chemistry are going to elicit a significant gill ionocyte response in the form of increased surface area and also microvilli. So our study question was, what is the magnitude and the duration of the stress response to soft release methodology? If we can determine the magnitude and the duration of the stress response, we can inform the optimal time of release from these enclosures for when delta smelt are at minimum stress levels. And hopefully if we're releasing fish at minimum stress levels, hopefully we're maximizing their post-release performance and survival. Okay, so this was my study design. We had 8 enclosures, soft release enclosures deployed close to an actual release site out in the delta. And we transported fish from FCCL out to this release location in carboys that were filled with either 200 or 300 fish. Then each carboy was emptied into enclosures such that each enclosure either had 200 or 300 fish. We also had one more enclosure deployed in a recirculating tank at FCCL to serve as a control for conditions in the field, and this enclosure was filled with 200 fish. So then we sampled fish for cortisol, glucose, and lactate and gill ionocytes at 10 time points across 4 days, mostly clustered towards directly after release on day 1. We also monitored latent mortality in each enclosure. So going to get into results quickly here. Basically, mortality was very, very low across all enclosures throughout the entire experiment, less than 3%. So we're not going to spend a lot of time on this. Quickly getting into what we saw with cortisol. Just to orient you to this figure, on the x-axis, we have hours after release. And this also includes our control group, which were fish that were sampled before any handling or transport had occurred. And on the y-axis, we have cortisol in the delta smelt, and this is expressed in nanograms of cortisol per gram of fish mass. So we saw no effect of density on cortisol. So the data that you're looking at here is pooled across those density treatments. And the blue color corresponds to data from the field location, and the pink color corresponds to data from the lab location, and the green is our control. So overall, what we saw was a significant peak from control levels in cortisol by around half an hour after release in the field location and the lab location. This is about a 60-fold increase in cortisol from control levels. This peak in cortisol was followed by a significant decrease in cortisol by around 27 hours after release, and then a further decrease in cortisol by around 72 hours after release. And in the lab, we saw a smaller peak in total cortisol and also a quicker time to recovery. For glucose, it's the same orientation of the figure as we just looked at for cortisol. And again, there was no effective density, so the data is pooled. And for glucose, what we saw was a significant peak in glucose in delta smelt from control levels in the field, but not in the lab. This peak in glucose was followed by a significant decrease by 27 hours, and then again, a further decrease by 72 hours after release. And in the lab, we saw a smaller peak in total glucose and also quicker recovery. Lactate, similar story, but we didn't see a statistically significant peak from control levels, but we did see an increase by around half an hour after release in fish in the field, followed by a significant decrease by around 6 hours. And again, in the lab, there was a smaller peak followed by quicker recovery. So this is our gill ionocyte surface area data. So on the x-axis, again, you have hours after release. We just analyzed gill ionocytes from subset of data points or subset of time points. And the y-axis, you have total gill ionocyte surface area. So this is the amount of the gill ionocyte that is facing the outward water and doing that ion transport that we talked about. So generally what we saw here is a significant increase in gill ionocyte surface area following release, especially in those high density cages. We saw an increase in low density cages, but just not as dramatically. So what's going on here? So we know that PCO2 was very high during transport, so acidity was high in that carboy transport water. And what we think this was eliciting was an ionocyte response to remove hydrogen protons and accumulate bicarbonate in the fish tissue to buffer the acidic conditions happening during transport. However, when fish were released to the field, PCO2 went back down and water chemistry returned to normal. But fish had all this, these delta smelt had all this built up bicarbonate that they needed to remove from their tissue, or else they were risking becoming alkaline. So we think that this increase in total ionocyte surface area after release is primarily due to the need for these fish to remove bicarbonate after they're released. So we saw that there was a significant peak in cortisol after release, and we also saw that there was a significant peak in total ionocyte surface area after release. And the literature tells us that cortisol regulates this ionocyte response. So this is just to say that we more or less confirmed that, showing that after this significant peak in cortisol, we saw this significant peak in total ionocyte surface area. At the beginning of the presentation, I also mentioned microvilli. So I just wanted to show you a picture of these microvilli and a picture of these ionocytes and explain them a little bit more. So the image you're looking at here is a picture of a gill ionocyte that we would use for analysis. And the ionocyte itself is this triangular structure in the center of the image. And these small features on the surface of the ionocyte are what we're calling type 1 microvilli, which are these kind of short, stubby features. Type 2 microvilli look different. They're these sprawling branching structures that kind of spread out over the surface of the ionocyte. And type 3 microvilli are also distinct. They're more tightly clustered and elongated, and are usually found to be surrounded by type 2 and type 1 microvilli. So our study is unique in the literature, and that is the very first to show all three types of these microvilli on the same apical area of a gill ionocyte. And these microvilli were expressed kind of differentially throughout the study. So the type 1 microvilli were used predominantly and throughout the entire study, mostly reflecting the same relationship as the total ionocyte surface area did. The type 2 microvilli, on the other hand, were used not so much at these early time points, but much more predominantly towards the 27 and 48 hour time points, and much more in those higher density cages. The type 3 microvilli were used predominantly 1 hour after release and still heightened at around 27 hours after release, but by 48 hours they had fallen and were being used less, especially in those low density cages. So we're not quite sure what's driving the differential use of these microvilli through time. But a postdoc in my lab, Garfield Kwan, is doing some immunohistochemistry right now to try to discern protein expression on these microvilli to try to figure out what's going on. So I just threw a ton of data at you, and I just wanted to wrap up very quickly. So in total, our cortisol, glucose, and lactate data tell us that supplementation is eliciting a considerable stress response in delta smelt. and that around 24 hours is needed for delta smelt to recover from these stress levels. Our gill ionocyte morphology data tells us that these shifts in water chemistry that are happening during transport and upon release are likely eliciting a significant ionocyte response. And this is represented by fish needing to expel that bicarbonate they built up. And this ionocyte response, along with the cortisol, glucose, and lactate and the generalized stress response altogether, is really just pulling energy away from nominal processes and functions and behaviors that the delta smelt would be needing to do to survive and function out in the wild. So altogether, delta smelt become very stressed during supplementation, but these soft release enclosures allow them a safe place to recover from that stress before they're released. Great. Thank you so much for listening. And I also wanted to thank my funders at the Department of Water Resources and all the scientists there who helped me out with my project. I also wanted to thank everybody at UC Davis and the Fungi Lab for their help with this work. Thank you. I think we have maybe time for questions. No? Oh, okay. Thank you. Sorry.
And our next speaker is Trishelle Tempel with the Department of Water Resources.
All right, thanks everybody. So as Denise mentioned, I'm Trishelle Tempel. I work for DWR. And today I'm going to talk about a recent delta smelt tagging study that we ran in collaboration with the U.S. Fish and Wildlife Service and the FCCL. Go ahead. Okay, sorry, same problem. All right, so first I wanted to give a little background on why we decided to look into this. And so as Katie mentioned in her talk, earlier this year, the ERTT wrapped up their third full season of delta smelt experimental releases. And the team released an impressive total of 91,468 fish. So one key component of this effort is that every single one of these fish were tagged with either a VIE tag or an adipose fin clip. And you can see both of those in the pictures here. So these tags enabled the monitoring survey field crews to instantly assess whether captured delta smelt came from the release effort. And on top of that, last season, each release event was assigned a unique tag type, which Katie also went over in her presentation. So this allowed the team to match every single recaptured fish to its specific release event. And this process really allowed the team to take a deeper look at the factors that might affect release success. So like Katie mentioned, things like differences in recapture rates between different release dates or different release methodologies. And overall, this is an immensely powerful tool that really helps the team evaluate and refine different release strategies in preparation for supplementation. But the downside is that these individual marks take a lot of time to apply. And last year alone, it's estimated that it took the tagging team over 850 total man hours to tag all the fish that were released in year 3. So what we wanted to do is to explore whether we could find an alternative tagging method that's suitable for delta smelt, but is also less cumbersome to implement. And in particular, we wanted to explore techniques that would be scalable to account for rising release numbers in years to come, techniques that would be easy to implement at the FCCL, and things that would still allow the team to uniquely mark different batches of fish. It was also really important that we weren't going to kill these sensitive fish and that this tag would last a long time. And so after reviewing the literature and picking the brains of a lot of folks, both in and out of state, we decided to test 3 new methods. We looked at photonic, ultraviolet grit spray, liquid nitrogen freeze branding, and thermal otolith marking paired with an adipose fin clip. So to conduct our study, we tagged a total of 1,050 fish in October of last year. 450 of those fish were treated with the photonic UV grit spray, and that comes in 3 different colors. 300 fish were marked using a liquid nitrogen freeze brand, and for that we tested 2 different symbols. 150 fish were VIE tagged, which allowed us to compare our new techniques to the current standard. And lastly, we tested using temperature fluctuations to impose an otolith mark onto 150 fish. And those fish all also received an adipose fin clip. That way we could still visually identify them. Once the tagging was complete, we divided all of our fish into 3 FCCL holding tanks where they remained for 5 months. And we didn't conduct any subsampling during the 5-month period, but we did assess the tags of all the mortalities along the way. And then after the 5 months were up, we pulled out all of our survivors and assessed their tags as well. And today I'm going to really focus on the first 3 methods listed here, the grit spray, freeze brand, and VIE. Those adult otolith marked fish haven't been processed yet, but I do want to note that Brian Alper from Levi Lewis's Otolith Geochemistry Lab has put together a really cool poster on larval otolith marking. So if you haven't already had a chance to check it out, I'd highly recommend taking a look today. All right, so diving into our tag applications, here is a closer look at how those VIE tags are applied. So for every single fish, a tagger takes a needle and injects that colored elastomer underneath the skin. So with this method, there's a lot of unique markings that are available because a tagger can precisely select where on the body to put the tag. And in our study, it took about 90 minutes to mark 150 fish, which gives us an average mark time of about 36 seconds per fish. So next I'll talk about our photonic UV grit spray. So to apply this mark, we would put 25 fish at a time into a black 5-gallon bucket, and then we net them into the mesh tagging frame that you can see in the picture here. So on the side, you can see Scott is holding a black canister, and that is actually a small sandblaster that's filled with a solution of yellow powdered grit and water. So to actually mark these fish, we would spread them out in this tagging frame and then go over their bodies with a 2 to 3 second pass with the sandblaster. Once they are nice and tagged, we dip them in this rinse bucket, and I wanted to play the video out so you can see they're okay. We'd give them a rinse, and then we'd net them into our holding tanks. So what the sandblaster is doing is essentially applying a tattoo. So what we're doing is embedding that pigment underneath the skin. And you can't see the pigment with the naked eye, but it does fluoresce under a black light. And it's not possible to get as many unique combinations because the tagger can't control where this spray is going on the body. But and you can also see that the application can be a little patchy. So some of those fish get quite a bit of pigment while others just get a couple spots. But this is something that can be applied really quickly. So in our study, it took us 64 minutes to tag all 450 fish, and that gives us an average tag time of about 9 seconds per fish, which is about 4 times faster than the VIE. All right, moving on to our freeze branding. So to apply these marks, we made the freeze brander that you can see in the picture below. And the way this works is liquid nitrogen is poured into that center pipe where it flows into those 2 arms you can see sticking out the side. At the end of each arm, we have a brass cap that's fitted with a 1/4 inch wide letter. And for our study, we tested 2 different letters. We looked at O's and V's, and there's a picture of each of those brands at the bottom. So before tagging, you have to cool the brander down for about an hour. But once the brander is nice and cold, the marking process is actually pretty simple. So what we would do is grab fish one at a time and then press them to that branding tip for about a second. And here's a look at what those brands look like following the tagging process. And as you can see in the pictures, some of these marks were really clear, others were harder to identify. And it took us about 90 minutes to tag 300 fish. So this gives us an average tag time of about 18 seconds per fish, which is about twice as fast as VIE. So overall, how did our methods compare? Going back to our initial needs here, we were able to find new methods that are scalable, easy to implement, and allow for differential tagging. But did the fish survive it, and did those tags last? So that's exactly what we're looking at in the box plot here. What we're seeing is the proportion of fish who both survived the full 5 months and had an identifiable tag at the end of it. And as you can see, our grit spray and our VIE groups showed pretty similar success rates, but these freeze brand groups, they fared a bit worse. So what happened? So first, we're going to look at survival. And the graph here is showing the ratio of our mortalities to our survivors for all of the fish that successfully retained a mark. And what we see is that survival rates were really pretty similar between all treatments. They're actually even slightly higher in the grit spray group, so it doesn't seem like survival is driving these differences. But if we look at our tag retention rates, it becomes clear that that's where the freeze branding is falling behind. So here we're looking at the tag retention rate for each of our methods. And as you can see, the VIE fish showed a really high tag retention rate of 99%, while The newer method showed some lower tag retention. We were looking at 80% retention for our grit spray and only 63% retention for our freeze branding. So when we look at tag retention over time, some pretty interesting patterns emerge. When we assessed each of these tags, we gave it a tag score, which was on a scale of 1 to 3. So tags that were identified as the score 3 were tags that would be really easy for a field crew to spot. They were really clear tags. Number 2s were things that would still be likely to be seen by a field crew, but they're a little less clear than the 3s. And a score of 1 are things that would be pretty difficult to find, probably a pretty easy miss in the field. So the plot here is showing us our average tag score across months. And at the top in blue, we see our VIE fish. And what this is showing is that VIE produced a consistently clear mark across the entire 5-month period. Moving to our grit spray fish in green, we see the peak tag scores happened in October. And this is indicating that the best tagged fish likely saw pretty high immediate mortality. But after October, those grit score tags were consistently visible, even though they were a bit less clear than the VIEs. When we move to our freeze brand fish in red, what we see is that these marks need some time to develop. So initially, the marks were pretty hard to detect, but the tag clarity sharply increased 1 to 2 weeks after application. But that clarity started to decline again in January, and it's not clear what's causing the decline. It could be that the marks are simply good for about 3 months, or it could be that these marks become less clear as those fish reach reproductive maturity. So as these fish mature, we start to see changes in their skin coloration and the skin texture, and that might be making these tags harder to spot. So speaking of fish maturity, unexpectedly, we saw a lot of fluorescent eggs in a lot of the fish that were removed at the end of the study period. So these eggs that were glowing, all appeared to be unhealthy ones, and they were detected in fish from all treatment types. So here we see an example of a grit sprayed fish up top and a couple of VIE fish below. And we don't know what caused the glow. Our first thought was that it could be related to the fish eating some residual grit particles while in their holding tanks. But these plastics are non-soluble, so we're not sure how they would have incorporated into the body, and this isn't something that they've seen in other species. So on the flip side, it's really not routine to blacklight and overripe fish like these ones are. So it could be that the glowing isn't related to the grit spray at all, and is instead somehow related to another factor that's influencing the atrophic condition of the eggs. But regardless, this was a pretty surprising find that we definitely need to look into further before determining whether grit spray is a viable option. So in conclusion, VIE tagging was our best performer, but it was also the most cumbersome to apply. It did show strong tag retentions, and we didn't see any unexpected effects on the fish. The freeze band tagging might be suitable for short-term marking, but the tag effectiveness may decrease after 3 months or as those fish enter spawning condition. And the UV grit spray showed some promising results, but we really need to investigate whether any byproducts in the holding tank are negatively affecting the eggs. So while we didn't find a method that can easily compete with all of the benefits of VIE, there are still some possible directions we could take the work going forward. So some potential ideas could be to investigate the cause of the decline in that freeze band clarity, investigate the cause of the mysterious glowing eggs, experiment with our grit spray methods to eliminate waste in our holding tanks, and continue to explore alternative non-visible tag types like otolith marking or genetics. And with that, I want to say a huge thank you to all the folks that helped pull this together, particularly to the FCCL staff who cared for our fish for a full 5 months, which I know was quite a big ask. And if I have any time, I can take a couple of questions. Okay.
I was curious if I understood you correctly that the fluorescent eggs were observed across all treatments?
They were. So all fish, all 4 of them, even the temperature fluctuation fish, they all had the fluorescent eggs, but all of those fish were divided evenly into our 3 holding tanks. So it could be unrelated to the grit. It could be something in the holding tanks that came off of the fish. It's just, we just can't tell right now.
Yeah, so biofluorescence is really widely distributed across fishes. So that's like reflecting light from the various tissues. So I'm happy to talk more about you with that.
Yeah, that'd be great.
It just seemed that the grid application would lend itself well to practice and improvement of abilities, and how long the fish were exposed to the grid could be looked at survival rates with 1-second applications or 10-second applications. And are you going to go down that road, or am I missing something?
We did it a little bit. We had to refine our grit spray methods a few times. The way you do it really influences survival. So we did have a high mortality rate when we were going a little too hard. So we did play with different pressures, different ratios of the spray to water in the canister. And this kind of seemed to be the sweet spot. You're kind of towing the line between survival and tag retention. So I think we can refine that balance, and I think we can improve on it, but we found something that worked for this first pass. Hopefully that answers your question.
One thing I'm wondering about is the potential for diet studies down the line to retain some of this. So like, do you anticipate that one of these is maybe not suitable, that maybe like it's in the stomach of another fish and the grit, for example, is wiped away or something like that?
Yeah, do you mean for the grit specifically? Is that what you're?
Yeah, I think that's probably the most relevant one.
Yeah, so we definitely need to investigate if this is getting incorporated into their bodies. It shouldn't be. So the grit spray is something that's done with salmonids really regularly over in Utah. And they've been doing it for years and years, 35 years or something like that. And they haven't seen any of these effects, but we need to first figure out if that spray is getting incorporated into the body. It shouldn't be, but.
So I was actually thinking like if it was being wiped away so that, like, let's say you were doing a diet study on a bass, you opened up the bass, you found the smelt, but now you can't tell what group it came from. So not so much for the bass, but for the smelt.
I hear what you're saying. I think that would be hard to do. So if we go back to a picture of what these sprayed fish look like. If I get there, okay. So what these are these tiny little particles in their body. And so you can see some fish only have like 1 or 2 specks. So I imagine as a fish is getting eaten in the belly of a bass, those are going to flush out pretty quick.
Thank you.
Thank you.
Okay, next we have Florian Mauduit with UC Davis.
Thank you, Denise. So I'm Florian Mauduit. I'm project scientist in non-fungus laboratory at UC Davis. And today I'm going to present you some work and how we strive to enhance delta smelt conservation, and in particular, how we are testing if rearing fish with a naturalistic approach is not only effective, but also feasible. Oh. Right? Yeah, that's what I do. Okay. Okay, so you all know that delta smelt population collapsed over the last few decades. And as a result, there are like tremendous efforts that are deployed to preserve the species from the extinction. And one of the milestones was reached in 1996 with the funding of the UC Davis Fish Conservation and Culture Laboratory and the development of a captive population. Fast forward to 2021, and we saw a major breakthrough with the initiation of the supplementation program and the release of captive reared organisms for the first time in the wild. So the implementation of supplementation program using captive reared organisms is often used as the last resort solution because it's a risky move. And so one of the main concerns is that when you have a captive population, you will always select for particular trait desired or favored by captive conditions. This is what we call domestication. So in short, no matter what you do, in captivity, you will always select fish that do best under captive conditions. And unfortunately, it's often not the same fish that would do best under natural or in the natural habitat. The second concern is the effect of the environment on the phenotype. So it's a bit harder to imagine. So I like to illustrate this by explaining that if you have like 2 clones or twins with the same genetic, if you place them in two different environments, it will result in person with two different personalities and behavior. So if we translate that to our delta smelt, you can imagine that rearing them under captive conditions will result in fish with different phenotype than fish that would be reared under natural conditions. So the risk if we produce fish and release fish that are not fit for the wild is that first to waste time and money because these fish will likely not survive when we will release them. And also it can be even worse if these fish breed with wild fish because it will decrease the fitness of the wild population and overall it will be just harmful. So conservation managers are well aware of the issue of domestication, and that's why FCCL has protocol and genetic managements to maintain a refuge population, replicating the wild population as closely as possible. For instance, they do a parentage analysis and they try to include wild-caught fish into their crossings to limit the effect of inbreedings. But it's harder and harder to include these fish. So after about 15 generations, they start to see some evidence of domestication. However, there is not much done on these second points. And basically, delta smelt are raised in conditions that favor their survival. So for instance, these fish are raised at a constant temperature of 16 degrees when they are young and under like a temperature of 12 degrees for the later life stages. They are also fed like multiple times a day with commercial food, and they never experience risk of predation. So you can understand that when we release these fish into the environment, they are maybe not fit for survival. So that's why our group is trying to see if raising fish like under more naturalistic conditions or conditions that are closer to natural conditions can help us produce fish with a better fitness and better chances of survival when we release them. So today I'm going to present the results of two studies. The first one was funded by the Proposition 1 and aimed at testing the effect of early life fluctuating environmental conditions on delta smelt fitness. And the second one was funded by the Metropolitan Water District of Southern California and aimed at testing if we could rear delta smelt in a naturalistic environment with minimal human intervention. So to begin with the first project, I'm giving you a bit of a background, and I'd like to explain that organisms can adjust their phenotype to environmental conditions experienced during ontogeny. So this is what we call developmental plasticity, and basically the environmental conditions that these organisms will experience during their first few days of life can affect their overall life. So what we wanted to do is try to unlock the potential of developmental plasticity to improve the fitness of delta smelt. So for this, we went to FCCL and they provided us with thousands of embryos, and we exposed them to from fertilization to hatch to different temperature treatments. The first one aimed at mimicking what we currently do to raise these fish. So they were exposed to a stable 16 degrees temperature from fertilization to hatch. The second one aimed at exposing embryos to controlled diurnal fluctuation between 16 and 20 degrees in our lab. And the third one aimed at exposing embryos to uncontrolled natural fluctuation by placing them in cages in a pond. So we let these embryos develop, and then we measured the hatching success and the thermal tolerance of the resulting larvae at 2 and 150 days post-hatch. So on this graph, you can see the temperatures that the embryos experienced during their development. So the fish from the 16-degree treatment were exposed to 16 degrees. The fish from the controlled fluctuation group were exposed to diurnal fluctuation between 16 and 20. And the fish that were in the pond experienced somewhat irregular fluctuation between 16 and 22.5. You can see on these figures that the temperature treatment didn't affect the hatching success of our embryos. We had hatching success around 90% for all the treatments. Then we measured the thermal tolerance of our larvae at 2 days post-hatch. So for that, we measured the heart rate of anesthetized larvae during a stepwise increase in temperature until arrhythmia. So in short, we recorded a video at each temperature, and then we processed each video with a software that calculates the number of beats per minute. And this allowed us to produce the plot that you can see at the bottom right that displays the heart rate on the y-axis that change with temperature on the x-axis. And you can see that for all treatment, heart rate increased with temperature as expected. But what's interesting is that fish that were exposed to a controlled fluctuation in our lab that you can see in red were able to maintain their heart rate a degree higher than fish that were raised in the pond and 2 degrees higher than the embryos that were exposed to 16 degrees. So in short, just by exposing the embryos to a controlled fluctuation for 10 days, we have been able to increase the thermal tolerance of the larvae by 2 degrees without affecting the hatching success. So that's cool, but remember that developmental plasticity can have long-lasting effects. So to test if it was like permanent, we raised these larvae for another 150 days under common garden condition at 16 degrees. And unfortunately, when we measured their thermal tolerance, at that time, we didn't see any difference. So this means that the difference we observed in larvae were no longer present at 150 days post-hatch. So now I'm jumping to the second study I wanted to talk about today. And in that study, we aimed to test if we could rear fish for at least part of their life cycle in impoundments without human intervention or a minimum of human intervention. So this is something that is done like in other successful supplementation programs, such as the Razorback Sucker and Rio Grande Silvery Minnow. And it has been shown to mitigate the effect of domestication. So we wanted to translate that, to see if we could translate that to a Delta Smelt supplementation program. So for that, we collaborated with Shona Cunha in the Metropolitan Water District, and we deployed 3 cages in 2 of their impoundments on Bolden Islands, and we named these impoundments East and West for our study. So we did 2 deployments, and each time, each cage had 60 sub-adults. So the first deployment lasted 6 weeks from mid-November 22 to mid-January 2023. And the second deployment lasted 13 weeks from February to May 2023. Fred Fryer's group from USGS monitored the temperature and water conditions during our 2 deployments. And you can see on this plot that during the first deployment, the temperature was between 8 and 16 degrees, which is well within Delta Smelt range of thermal tolerance. And so as a result, we saw an exceptional survival at the end of the 6 weeks deployment with survival over 95% overall. Also, we saw that in the East impoundment, fish were able to grow by 40% of their initial body mass, but on the West impoundment, they didn't grow. We believe that it's due to the warmer conditions the fish experienced in the East pond at the beginning of the deployment. During the second deployment, temperature got warmer. We started with a temperature around 8 degrees, and we ended up when the temperature reached close to 24 degrees. And so since it was a longer period, we did 2 sampling points, one after 6 weeks and one at the end of the 13 weeks period. So 6 weeks post-deployment, we saw a survival rate over 80% in both impoundments. And 13 weeks, we had a difference. So it was close to, still close to 80% in the East impoundments, but we observed some mortality in the West pond. What's interesting is that at the same time in the West pond, we saw a lot of eggs like in the enclosures. So we believe that the mortality was actually some post-spawning mortality because Delta Smelt are known to die after spawning. And so on the growth chart, you can also see that fish grew by 30 to 40% of their body mass during that period. So in short, in that study, we showed that Delta smelt subadults can survive and grow in cages in the impoundments from at least November to May. So to conclude, in these studies, we showed that it is possible to improve Delta smelt thermal tolerance by exposing them to fluctuating temperature during their embryo development. So this has the potential to increase the fitness of the refuge population and mitigate the effect of domestication. We could also use that to better prepare the early life stage for natural environmental conditions in the event of early life stage release. Our group is also trying to see if we could expose captive reared fish to predators to develop their predator avoidance and better prepare them for the wild conditions. In the second project, we also showed that Delta smelt can be raised for part of their life cycle in enclosures placed in impoundments. So we could raise Delta smelt under these conditions and mitigate the effect of domestication like it's done in other successful supplementation program. We could also use that as an alternative to hard and soft release by transferring fish for a couple months into these impoundments before releasing them in the wild. And importantly, it also has the potential to reduce the manpower and space needed to raise them, because like raising subadult Delta smelt takes a lot of time and space. So we could see in the future that FCCL focus on early life stages and the refuge population, and that all the fish for supplementation and research could be reared in these impoundments, and that could also help us to increase the number of Delta smelt produced each year. Thank you for your attention. We have time for one question.
Great. Thank you. Excellent talk. Forgive me, my training in this area is a bit dated. Can you speak a little bit about the evaluation of fitness? Is that best thought of as an individual trait or a population trait? And how would that affect your strategies for supplementation?
So I measure individual fitness. So we believe that temperature tolerance is one of the important determinant of fish fitness, which is defined as their ability to survive and transmit their genes. So yes, for now, we measured fitness at the individual level, but we believe that if we release individuals with a better fitness, the population overall will have a better fitness. Thank you.
All right, thank you very much, everyone. Really cool stuff on experimental release. Now we're up for my favorite part, which is the poetry slam. So if you would like to pitch your poster, either through prose, poems, song, skit, etc., please stand up and line up in this hall here. And I really hope it's not just me, because some people were like, oh, should we do a poetry slam? I don't know. And I said, come on, this is the best part. So if I'm going to be left hanging, I'm going to be really embarrassed. Oh, we have a guitar. Oh, I'm excited. Good, good. All right, I'll head up first here, so you don't feel shy, and then we'll just go in the order of people standing up by the back wall. So we're here at the IEP workshop. Our science, we all know, is tip-top. With nutrients, fishes, zooplankton, and wishes, the fun and the learning just won't stop. So sit back, relax, and enjoy while you learn about smelt in carboys, or Phragmites control, or wetlands with soul, and hang out with our girls and boys. We may all get tired of meetings and Facebook and TikTok and tweeting, but now we're together and it only gets better. IEP is awesome and it bears repeating.
Hi, everyone. My name is Helen, and I'm come all the way from Boston because I have some research to share that's really pretty awesome. You could say it's cool, but then you'll be a fool because science needs to matter to the fish. So we're trying to figure out the risk that's hiding in the Delta smeltless prey from the contaminants in the Delta waterway. We gathered a large sampling crew and went to the Sacramento River and Cashleu, and we collected amphipods, insects, and zooplankton. So many different species, it was nearly a ton. We wanted to know if the pollution is causing the invertebrates to undergo evolution. To do this, we sequenced their genes, and we found out that it means that they can become resistant to insecticides, but for nature, this is not wise. Because now there is poison in the food web that got mobilized out of the sediment bed, and it ends up in the fish we care so much about. You want to know how? Come by my poster to find out. Poster 8D is where you will find me.
Okay, we have a couple posters from the USGS biogeochemistry team with all our collaborators on our high resolution boat-based mapping, both in channels, shallow wetlands, and then also how we use that data to inform our remote sensing algorithms. So here we go. The BGC group likes to go mapping. It's a place where everything is happening. Finding phytoplankton in channels, measuring nutrients in canals, new instruments and approaches are inspiring. We measure things from the ground, air, and space, working on boats in a beautiful and interesting place. We are game to try anything, providing data for modeling. Our complex, challenging delta requires we keep up a frenetic pace.
Hi, my name's Kat. I just have a quick haiku. Ephemeral blooms. There could be toxins in that. You might need a spat.
I did this one time like 10 years ago, and now I have to do it every year. My name is Brendan Lehman. I'm with the Southwest Fisheries Science Center in Santa Cruz, and I don't have a poster, but I'm giving a talk tomorrow afternoon. And this song's not about that either. It's about a website you can go on and track fish that are acoustically tagged in the Central Valley in real time that my team developed. Oh, fishy, where did you go? I'm asking for scientists who are wanting to know, where do they swim and where do they sink? Can we figure it out before they go extinct? We caught them and tagged them and set them all free. You can track them in real time on the internet, indeed. It's as simple as typing in the command bar above. oceanview.pfg.noaa.gov.
All right, thank you so much to everyone who had the guts to come up here and share your poetry and songs. Hopefully that inspires the rest of you to come up with some haikus, limericks, ditties for next year. I know it's been a little while since we've had a real poetry slam, so trying to get that started back up. So now we are going to transition to a short break. Please enjoy the coffee outside sponsored by ICF. Immediately after the break, wander upstairs to the poster room. We're going to have our poster session, followed by lunch. So a good long chunk of time where you can pretty much do whatever you want. But definitely emphasis on all the amazing posters up there. I spent 2 hours up there and still maybe only saw a third of the posters. So enjoy, come back here at 2:10 for this afternoon session. And lunch, you have an hour and a half for lunch because if you need to leave the building and find some food, it can take a little while. If you scan the QR code for the workshop website and go to the area guide, has some recommendations for restaurants. All right, enjoy, and I'll see you back here at 2 o'clock.
If anyone has any presentations to load, please stay here and let's do that now. Thanks.
Thanks. Sorry, forgot that. For coming back after lunch. We have a couple more great sessions for the end of today, and then we have all day tomorrow. So lots more fun to be had. So without further ado, I'm going to hand it over to Nicole Kwan, who's going to introduce our next session on floodplains, wetlands, and restoration.
Yeah, Rosie pretty much took the words right out of my mouth. Next up, we have wetlands and floodplains advancements in monitoring and restoration. So thank you everyone for attending. Our first presentation will be from Dennis Finger about acoustic telemetry work in the Yolo bypass. So let me just cue that up. All right.
Hi, everyone. My name is Dennis Finger. I'm an environmental scientist with the California Department of Water Resources. And today, in an ode to Dr. Seuss, I'll be talking about the places they'll go. Talking about results from a study on acoustic telemetry for adult salmon and sturgeon in the Yolo bypass. The Yolo bypass is a flood conveyance basin that provides vital floodplain habitat for native fish throughout the Delta region. It's a very important migratory corridor for listed salmonids and sturgeon passing through the area, where salmonids and sturgeon will be coming from downstream, either the estuary or the ocean, and will be passing through to reach spawning grounds further upstream. And in part because of this status as being a pretty important region for habitat, there have been a number of habitat restoration projects throughout the Yolo bypass in recent years to improve rearing and to facilitate fish passage. And does that work? Okay. Some of these projects, notably to improve rearing, the Big Notch project, which will be coming online shortly, will reactivate floodplain habitat throughout the Yolo bypass. It also facilitates passage, as does the adult fish passage, both of which allow fish to get through Fremont Weir. The agricultural road crossing projects that improve previous barriers that were in place, and Wallace Weir that helps fish get back to the Sacramento River. But with all this habitat restoration in place, as well as all these important fish passing through the area, it's really important to have data to help us understand these barriers and evaluate the restoration projects. And that's where acoustic telemetry comes in. So we lead an acoustic telemetry study, and basically how that works is we catch a fish, we put a tag on it, the tag gives off a unique sonar pulse, and we have receivers in place that will pick up that exact detection at a certain date and time. So in that way we can track fish movements throughout a given area. And we do this in the Yolo bypass to catch and tag white sturgeon and fall run Chinook salmon, where white sturgeon help us to understand the threatened green sturgeon, and fall run help us to understand threatened spring run and endangered winter run salmon movements through this area. And this is an enormous collaborative process where we have to have a ton of help to do so, partly from DWR's Aquatic Ecology Unit, our own team, and then a number of volunteers from DWR and CDFW that help with quite a bit of this field work. It can get intensive. This is drone imagery that JT in the crowd captured. So this is how we'll catch salmon via gill netting. And over the last, so I'm talking about our study over the last couple of years, and we've done quite well over the last couple of years, catching and tagging about 50 salmon each in 2022 and 2023, and then 5 sturgeon in 2023 as well. So with all these tagged fish, we have a few questions that we hope to address. The first of which is, do known passage barriers in the Yolo bypass affect salmon migration? Secondly, we sought out to address which directions do most salmon move within the bypass? And then third is, where do salmon and sturgeon go after leaving the bypass? And so for this first question about how do known passage barriers affect salmon migration, we looked at residence time, so the amount of fish that the amount of time that fish are spending both downstream and upstream of barriers. And I got some help on this from Rosie Hartman. And so this graph shows on the y-axis a log of residence time, so the amount of time that fish are detected at these receivers on average. And then on the x-axis, we have a few known barriers within the Yolo bypass, including Lisbon Weir, the Los Rios Check Dam, and Agricultural Road Crossing 4. And these bars indicate differences between residence time downstream versus upstream of barriers. And we're able to see that particularly Lisbon Weir, there's a significant difference where fish were spending more time on the downstream side. So they were getting perhaps blocked and unable to move upstream. And even at the other stations where we did see some difference, and overall, it's notable that there are still barriers and it's important to continue doing restoration. And this Agricultural Road Crossing 4, there's a project that will be coming along in not too long. Our second question was looking at salmon movement within the Yolo bypass. And this is, again, a map of the Yolo bypass with a red dot showing our telemetry array kind of spanning north and south within the Yolo bypass. We tag salmon right around Lisbon Weir, indicated by this triangle. And once we tag fish, they can go a few different directions. So some fish will go to the north, where they can get to Fremont Weir, to Wallace Weir, where they can get back to the river that way. And this is a track of a fish that we saw doing that, where on the y-axis, we have, these are different receivers that we have going from the southern end to the northern end, and on the x-axis is time. And so we see that this fish, for instance, kind of moved up and down within the system for a little bit before going to the northern end. Another direction fish could go is down to the south, and where there's the ability to exit through the southern end of the bypass and then potentially find spawning grounds in another system. This is a track of the fish that we had that displayed that movement pattern, and so kind of hung around in the central part of the Yolo bypass for a while, and then all of a sudden moved towards the southern end. We saw a number of fish that hang out in the central part of the bypass as well, that kind of moved back and forth, but we didn't detect them either moving to the north or to the south. And then finally, this is perhaps the best outcome for fish that come into the Yolo bypass, is they could go up Buda Creek, where there's abundant spawning grounds that has been the focus of restoration efforts within the last couple decades. And this table here summarizes the last detections, or the last place where we saw some of these fish. So to summarize kind of broad trends, we saw that about 10 between both years combined ended up in the northern part of the Yolo bypass. And so some of these fish were able to exit via Wallace Weir. Others could have just been gotten lost within the system in the northern area. About 10 combined between both years ended up going up Buda Creek, and we know that it's been a banner year for Buda Creek salmon returns in both years, so it makes sense that a number were able to go up that way. a number we're seeing for the last time in the central yellow bypass, which is not a great outcome. And it's possible that we know that some mortality happened where fish weren't able to go any other direction, but there's also a lot of tag shedding that could take place, which could inflate those numbers a little bit. And then finally, we saw that number we're seen for the last time in the southern part of the Yolo bypass, where they could have thus exited the system and gone to other areas. Speaking of other areas, our last question is looking at movement beyond the Yolo bypass. And for this, it was, again, a collaborative effort. So we have receivers within the Yolo bypass, but other groups have receivers in other places. And we've been able to do this analysis thanks to interagency working groups and data sharing from a number of different groups, including UC Davis, NOAA, DWR Feather River Program, USGS, US Fish and Wildlife Service, and many others as well. But with this combined data, pretty cool. We're able to see a pretty wide range of places where fish have gone. So this image here is a track of a sturgeon. So we tagged it here in the southern part of the Yolo bypass. And then these other dots show receivers that other groups maintain. And we're able to see a temporal movement over the course of about 6 months, where we had the sturgeon that we tagged in May 2023, and we last saw it on January 1st, 2024 in the Sassoon Bay. So it took a while for this movement to happen, but sturgeon are longer lived. They, when we're tagging them, they're not necessarily towards the end of their lifespan, so they can hang out in Sassoon Bay. Whereas for this salmon, for instance, that we saw, we tagged it around November 5th. And then by November 9th or November 10th, it had moved all the way down from the Yolo bypass, up the Sacramento River, up the Feather River, and then finally was last seen in the Yuba River just 6 or 7 days after we initially tagged it. So traveled quite a large distance in not very much time. And so this figure shows just a map of all the places where we saw fish turn out, with the larger dots being places where we detected more fish, and the large dots are all within the Yolo bypass, but quite a large range of locations where we saw these fish turn up. And looking again at a table of final detections, we saw a wide range. So again, a number of fish were seen for the last time in the Yolo bypass, especially for salmon. But a number, as we mentioned before, ended up going up Puda Creek. There are a number that kind of went towards the southeastern part of the system. So either to Sassoon Bay, or one that even went out to the Golden Gate, and then one that went out to the San Joaquin River. So went pretty far to the southeast. But a number of these fish ended up in productive spawning habitat. So that could be in the Sacramento River, in the American River, in the Feather River, and in the Yuba River for salmon. And then for sturgeon, again, there's something of a range. We saw more so that a lot of these sturgeon ended up in Sassoon Bay, but some of them did go up the Sacramento River as well. So to kind of summarize a little bit, we saw overall that fish were spending more time downstream of barriers than they were spending upstream. So they're potentially getting blocked. There were many salmon that within the bypass that did move towards spawning habitat, though there were also a number that did not. Another ode to Dr. Seuss. We saw a number of places that fish went. So there's a wide locational range of last detections, particularly for salmon, but also for sturgeon as well. And then moving into the future, with the Big Notch projects coming online and other restoration projects that will continue, these will continue to improve the migratory corridor through the Yolo bypass. And it's important to continue our monitoring, doing studies like the one that we're doing to see how these processes change the area. With that, thank you for your time. Okay, yeah, we've got time for questions.
We have one online.
Yeah, for one of our remote participants, they asked, Dennis, do you think poaching or fishing harvest in the bypass may have increased the number of salmon last observed in the middle portion of the bypass?
Yeah, that's a good question about whether or not poaching could increase the number last seen in the central part of the Yolo bypass. And it's certainly possible, but I think it's less likely this past year in 2023 when the salmon season was totally closed. And so I would imagine poaching to be a little bit less likely. It's definitely a possibility though. I think I do imagine that tag shedding is probably a big reason why those numbers are so high, but that's definitely something interesting to look into a bit more. Yeah.
You don't have any, it looked like in one of the slides, you had receivers along the Sacramento River, but that one ended up down out of the bypass and all the way up to Kwan in the other place. Didn't have any docks located over there. Are the receivers over there?
I think maybe this is the slide you're talking about.
Yeah, so you have records from the bypass and you said it went out the bottom of the bypass, then up the Sacramento River, but you didn't, there's no recognitions of it. But then on another side, you had receivers located along the river there. Could it get by that many receivers? And
yeah, that's a good question, and that's an array that's from another group that we don't maintain, so I can't speak to the coverage within that area. It's certainly possible that it could have passed by some of those receivers, and I don't know, as we're seeing here, is swimming pretty quickly, and so like may not have been opportunity for.
But it rang all the bells on once it got to the north.
Yeah,
just seems weird. And any thoughts on why you caught one? Why the last capture of one was at the Golden Gate? That seems hard to understand.
Yeah, it's a good question. I don't know if I have a great answer for that either. We did see, yeah, there was the last track. I think it might have been picked up at Point Bonita, like outside of the Golden Gate as well. And speculation is possibly it could have aborted its spawning run and headed out, or it could have just gotten very lost. I'm not totally sure. Good question to ask.
Thanks, Dennis. Next up, we have Stephen Deverell talking about floating peat wetlands.
Thank you. Thank you for the opportunity to speak today. I appreciate the opportunity to talk about some kind of unique ideas relative to restoration in the Delta. We've been looking at this floating peat problem for a while. Okay, I'll do that.
Just first time.
Thank you. So the question might be in your mind, why concern ourselves with floating peats in the Delta? And just from a regulatory standpoint, we know that tidal wetland habitat is integral to the co-equal goals, and the regulatory requirements actually mandate that. And we also know that the loss of tidal wetlands was tremendous with development of the Delta in the mid-19th century. Reduced the ecosystem net primary productivity by as much as 94%. But subsidence limits tidal marsh development in the Delta in its current state. This is just a photo on Twitchell Island looking up at the San Joaquin River. We're about 23 feet below sea level. And it's hard to put tidal wetlands in a place like this. We did, I'm reminded today that about 30 years ago, we started mesocosm experiments. We're looking at accretion of non-tidal wetlands in the Delta. And that led to the development of the West Pond on Twitchell Island, in which we've seen about 60 centimeters of accretion over the first 20 years there. So it's about 3 centimeters per year in this wetland that's still there on Twitchell Island. It's been surrounded by other wetlands. But it was the first restoration of non-tidal impounded marsh in the Delta for subsidence reversal and carbon sequestration. But as you look at the opportunities for creating tidal wetlands and accreting to tidal range with these non-tidal wetlands, there aren't very many opportunities. As you can see in the pink areas here, we're looking at less than 50 years for the most part. But as you go into the Central Delta, where you've seen the green and the blue, we're talking about 100+ years for accretion to occur to the tidal range. So there isn't much of a possibility that non-tidal managed marsh is going to lead to any kind of tidal wetland restoration. And we pursued this a little bit further in a recent article that's about to be published in San Francisco Estuary and Watershed Science, where we tried to look at land covers for greenhouse gas emissions reduction and restoration. We really wanted to ask the question, how can the configuration of wetland restoration and rice cultivation be optimized for greenhouse gas mitigation and restoration? And so you can see here that the maximum, as we defined it, the maximum potential restoration is really this situation where we have a lot of non-tidal wetland, this managed marsh, and about in the 300,000 acres that used to be occupied by tidal wetland. And around the periphery, the blue area is the tidal wetland that could be created within a reasonable period of time in light of sea level rise. So the question is, and so there are other issues too. The risk of levee failure and island flooding is there. Studies that have been done indicate some multiple island flooding in the future. Flooded islands such as Mildred Island and Frank's Track are not great habitat for native species and have high proportions of alien species. And construction of tidal wetlands requires a lot of fill material. So the feasibility of creating tidal wetlands on subsided islands is really small. So we've been looking at this idea of maybe substituting floating wetlands for tidal wetlands for a while. It came up, the idea has been floating around for a while. And we started this mesocosm experiment on Bolden Island in 2019. In the northern, actually adjacent to the McCollum River, we put in these 8 doughboy pools, which we're calling mesocosms. They're supplied with water from the McCollum River. And so we were able to gather some peat blocks from Twitchell Island, that 1997 West Pond wetland that I mentioned earlier, and place them in these mesocosms. And you can see in July, they were devegetated so we could move them. We moved them from Twitchell to Bolden and then placed them in the pools, and they grew pretty fast. Within September, they were fully vegetated. And this January 2024 is what they looked like recently. They're actually willow trees growing on them, and they've expanded substantially. I'll talk a little bit more about that. So as we conducted this experiment, we wanted to evaluate the continued flotation, food web benefit, biomass accretion, greenhouse gas emissions and removals, and water quality. We looked at 2 treatments, one in which we put, we filled the space of the pool with half the space was filled with floating peats, and the other treatment was it was 100% covered. The other was looking at varying residence times. We had a short residence time of 5 days and a long residence time of 10 days for the water in these mesocosms. We didn't really see an effect of that treatment on any of these aspects that we were looking at, but except for water quality, where we saw lower methane concentrations associated with the lower residence time. So greater movement through the pond or through the mesocosm resulted in lower methane concentrations. Carson Jeffries and his team at
the Center for Watershed Science looked at zooplankton and macroinvertebrates. I'll just talk about zooplankton a little bit today. And he did it for a year. This is about a 3-year experiment. I was able to see increasing zooplankton populations or density with time relative to the inlet, which is, you can see here, I'll show in the next slide. They were primarily cladosterans, but there are other species as well. And in general, we had about 3 times the zooplankton density in the mesocosms relative to the Mokelumne River. There was another aspect of these floating peats that it was somewhat intriguing, and it was based on some work that de Moraes did in Czechoslovakia, where he really looked at this root complexity as a benefit to fish for a number of reasons. One, it provided refugia, lower light, and he compared the fish captured in the root structure associated with floating wetlands versus the control, and there were substantially more fish captured associated with these complex roots in the floating wetlands. Also, he stated that biofilm may provide food for zooplankton. So there's some evidence that root complexity could be a benefit for fish habitat. The blocks accreted both horizontally and vertically. We saw about 5 to 9 centimeters of accretion, consistent generally with the Twitchell Island wetland accretion that we've measured. But we did see more than 3 square meters of horizontal expansion in the treatment where we only had 50% of the area covered with peat blocks. And the peat blocks grew together, so it's really formed substantially solid masses in these mesocosms. The methane emissions were generally consistent with what we've measured in other wetlands in the Delta. Most of these data are in publication by Hemis et al., documenting the data that's been collected by the UC Berkeley Baldacci lab. The floating peats were a little bit lower, but I'm not sure we can really compare the methods are somewhat different. But the net ecosystem carbon balance was greater than the other wetlands. I suspect that's because there's this abundance of water and nutrients in the system that really can allow a lot of carbon sequestration. But preliminarily, it looks like a net greenhouse gas benefit. So what are the next steps? I go on to that at this point. What do we do with this? I think we've run out of time to work with the mesocosms. Thank you. So just to talk a little bit about that, but also show you a picture of what we're doing, what we've done recently. We cut the vegetation down in 4 of the pools, put a cargo net underneath some of these blocks, and moved them over to a pond on Bolden Island. This is Mark Olds from Hydrofocus walking on this block of peat. You can kind of see it floating up and down. And then this shows us moving the same block actually into a trailer to take it out to the West Pond. So you get a sense of what these things look like underneath. They're pretty massive. A lot of biomass is accumulated here. They were about a foot thick when we put them in. And then here's what they looked like on March 15th in the West Pond. This is an excavated pond on Bolden Island that was used for excavating levee material. One of the things that MWD arranged for this, and these photos, these last two photos are courtesy of Russ Ryan at Metropolitan Water District, but the smelt cages are out here. So the idea was to look at the possible effect of the food web that might be transferred into the smelt cages from these floating wetlands that are out there. The wind direction is to to the east, so they're immediately downwind of the floating wetlands. This is a April 12th picture that shows the vegetation, but it also shows, if you can see in the background there, the net pins or the cages for the smelt that are quite a ways away, which would be the control. So the idea would be to look at fish survival in these two situations. One went close to the floating wetlands and one a ways away. And this is a picture that was taken April 10th. You can see the vegetation starting to come in already, and they're staying in place. So that's kind of where I'll stop today and entertain any questions. Thank you for your attention today. I appreciate also the funding from Metropolitan Water District, cooperation with the Department of Water Resources, and many others who participated in this project. Thank you.
Thanks. Do you get, I don't know if you looked for this, but do you get any fish associated with these floating peat islands in the periphery or underneath them?
Can you repeat the question?
Do you get any fish associated with these islands around the periphery or underneath them?
Oh, these are problems?
Well, in your last photograph, you had a floating island. It didn't. Right.
Yeah, we're not really equipped to do that. I didn't see to look at survival in the actual cages itself. The place adjacent.
Can you hear me okay? Okay. This is a fascinating project. What does it look like scaled up? Like, are these just going to float around Frank's track? Or are you going to like tie them down to certain areas? Or
Yeah, good question. And we're giving some thought to this. I think the first next step is to see how these behave in an open water where there's turbulence due to the wind and such, and see how they hold up. My prediction would be that they continue to grow and continue to thrive. I think eventually, and I appreciated the presentation on the setback levees, but this might be a next step. It's to try these floating wetlands and a setback levee where they could provide refugia as well as a food web benefit. Ultimately, actually, I've got an extra slide here of what scaling up might look like. Well, here's the setback levee where they might be a good place, such as the Twitchell Island setback levee. But as part of the Delta Islands adaptation strategy, this light blue area on Bolden Island is a place we've thought where you could put a setback levee or some kind of barrier that would allow you to create these floating wetlands in areas that have some kind of cross levee, such as this light blue area on what we call the Rabbit's Foot, down towards the southwest of the island. So these are ideas that we've been thinking about, but it's going to take a few steps to get there, I think.
All right, last up, we have Julian Wood talking to us about birds. So.
Hi, everyone. Thanks for sticking out for the last talk. And yeah, about birds, no less. You know, you've probably heard enough about fish, right? Hands up for birds. Okay. So my name is Julian Wood. I'm the San Francisco Bay Program Leader with Point Blue Conservation Science. And I'm going to be talking, this is basically like a methods comparison, so preliminary results. And this is actually phase 2 study. Phase 1 was focused on, resulted in creating what we call the Delta multiple benefits framework. And so that is a flexible science-based framework for conservation planning in the Delta, and that incorporates a multiple benefits approach to decision making. And it encompassed many different habitats, but did not include tidal wetlands habitat and tidal wetlands birds. So phase 2, we're filling that gap. So this is going to be about tidal wetlands and also developing tidal wetlands restoration scenarios that might include the floating peat islands. It's a pretty promising idea there. Anyway, so that's just kind of an overview of what I'm, the context and what I'm going to present. I do want to thank my co-authors, Christy here. She's kind of led that phase 1 approach and led the modeling efforts. And Megan kind of spearheaded the field methods, the field effort, and Hillary and Rose helped out with the ARUs, the ARU analysis. So my key messages: the San Francisco Bay Delta wetlands provide excellent habitat for a diversity of birds, and I'll show you some of those birds soon. And combining traditional surveys and ARUs greatly increases our ability to assess biodiversity. So it is a really good tool. ARUs are more cost-effective in detecting and specifically less common and secretive marsh bird species that are more difficult to pick up in our traditional point counts. And finally, like I mentioned, these results are going to feed into habitat and scenario modeling to ultimately improve tidal ecosystem restoration decisions for the Delta via this Delta multiple benefits framework. For those of you that aren't familiar with Point Blue Conservation Science, we were founded as Point Reyes Bird Observatory, and we're still rooted deeply in bird ecology and bird conservation, but have expanded to other disciplines and other areas since then. And I'm going to try to make this quick, so I only have 10 minutes or so. In brief, our study area is the San Francisco Bay Delta and Sassoon. So I'll show you some maps of the exact study locations, but that's what I'm going to be focusing today. And in terms of the habitats, like I mentioned, tidal wetlands, tidal marsh specifically, and also tidal riparian. And you all probably know that that's kind of a gray area there. There's a gray line between those two habitat types. We're covering both, as well as the open water, including submerged aquatic vegetation. And so some of the birds that are associated with these habitat types, these are our focal species, 7 of these. I don't have time to talk about each one of these, but I do want to mention the common yellowthroat and the song sparrow both have subspecies that are endemic to, in this case, Sassoon, and are bird species of special concern due to low population size. And then the black rail is also a focus for this particular presentation too, being a state threatened bird species and extremely hard to see and almost just as hard to detect, to hear. So the focus is on tidal wetlands, and you've already heard, I'm sure, before the tremendous loss for this in the Delta, and definitely hopeful that this goal. Well, I should back up and say, like 97% loss of tidal wetlands. It's kind of hard to wrap your brain around, and especially amazing that many of these species are still persisting in this area, even with only 3% of that habitat remaining. So that's pretty impressive, and some of the fish too. And so definitely hope that these species can not just persist but thrive as we work towards that goal of restoring more habitat. Okay, jumping into the field methods. So again, kind of focused on these two kind of comparison of these two methods, the passive point count surveys. So these are just 5-minute passive, meaning no called broadcasts used surveys, both in Sassoon and the Delta, and then the ARU, the automated recording units deployment. And that occurred only in the Delta, but overlap at the same locations as the point counts. And we'll see what that looks like. First, kind of go through the point count surveys and some quick results. Then we'll look at the ARUs and some quick results, and then a comparison. So that's where we're going. So here's a map of the point count locations. Kind of, you can see they're kind of clustered in the central Delta. And we did a data gap analysis just to make sure that, because we're ultimately going to be doing the bird habitat analysis here. And so we wanted to make sure that all the habitats that we will need to model are captured in these point counts. So we did a data gap analysis and found and added some sites based on that gap analysis. So we were missing some characteristics in the East Delta there. So we added those point counts last year, as well as this Liberty Island and the Goodyear Slough, which unfortunately has a lot of Phragmites. So that's point counts. So point counts results. So this is looking at presence, absence, and color coded the birds. We're just looking at these three here: least bittern, black rail, and yellow-breasted chat. So these are the ones that are less common, hard to detect. And so we really wanted to know what was going on with these birds. So just at first glance, you can see not that many black rail detections, only a few different spot red dots there. They were still surprising to get. I was happy that we detected those at the time, but yeah, it's not very many. And the same with chats, only a few. So that's point counts. And that's just looking at last year. If we included past years, we get a few more dots, but not that many more dots. So the ARU, real quick on the deployment technique. These have been around a while, but still it hasn't, there isn't a ton of information how to successfully employ these in the field, especially in as crazy a habitat as the Delta wetlands. But yeah, so we placed the units on long PVC poles that I spray painted camouflage to deter theft, which worked. None were stolen, amazingly. Still can't believe it. And planted the poles pretty much at the edge of the vegetation in the channel. And that was a trick. If we were lucky, yeah, you could get the boat, kind of a flat bottom, little Zodiac boat, get right up there and just plant them. But sometimes even chest waders would not have been sufficient because this is some pretty deep water and you can only get so close. So yeah, the wetsuit is where it's at. I can see why there's not a lot of fish studies like in the interior of these marshy habitats. It's pretty hard to get around in there. Sometimes you can get lucky and walk on the submerged aquatic, on the floating vegetation. It's kind of risky though, sometimes fall through. Again, the wetsuit. That's my take home message, wear a wetsuit in this habitat. Okay. And then we'll circled that little, you can see the ARU kind of deployed there. Sort of hard to see. So we collected 7,000 hours of audio data from these ARUs. So a tremendous amount of data, more than we can go through, right? Just by listening and identifying birds. So we used the BirdNet analyzer, and this is out of Cornell. This is the same algorithm that powers the Merlin app, which I don't know, hands up if anyone have heard of that app. Yeah, it's a great app. Just turn it on and have it listen, and it'll tell you what birds are in your backyard or wherever you are. It's pretty cool. So we use that same algorithm to identify these birds that we're looking for. We tested multiple settings to try to optimize the true to false, the ratio of true to false positives. And then each of the audio that came back as a positive for a given species and point, we verified. So we actually, me and my co-authors listened to it to make sure, yep, that's correct. Because as you know, all the Merlin users, sometimes it gets it wrong. So you got to make sure. Okay, results from the ARUs. So now we're just looking at a subset of those kind of core Delta sites. And all of these are point counts. There's a few point counts that we couldn't get the ARUs in there for different reasons, but. So right away, you can see a lot more dots, right? A lot of those red dots, the black rails, super exciting. This was actually a surprise to me. I really did not expect there to be this many of these birds, that we had missed that many of those birds. And especially in, oh, it's already circled. In this area circled here in blue, there were some historic surveys done for specifically for black rail, like employing the broadcast. And that area was surveyed, but no black rails were detected. So pretty unlikely that they were missed on those surveys. So I'm assuming that those rails actually moved into that area, and that's a new spot for them now. And Disappointment Slough. It's exciting. So now looking at the point count versus the ARU. So this is the test here. So this is, again, this is looking at prevalence. So the number of points where the given species was detected. And you can see the top three that we were just looking at: least bittern, black rail, and the yellow-breasted chat. Definitely more detections with the ARUs. More points had those birds were detected versus the green point counts. As you get a little lower, and also the American bittern definitely picked up a lot more of those, just because they don't call all that frequently. And so they were missed on our point counts. Oh, and I should mention, so the ARUs per point, we're talking about 7,000 minutes of audio per point. So that's what's represented here versus the point counts, 10 minutes per point. It's 5-minute point count, visited twice, 10 minutes, that's it. You're there, you're gone. So that explains a lot of why we're picking up all these other birds, just the sheer number of minutes that are out there recording. Lower down, the common yellowthroat, the marsh wren, and the song sparrow. So those were, more consistently picked up by both methods. So if those are your study species and you really want abundance, which you can't really get with the ARUs, right? That's more presence absence. Then maybe the ARUs don't make as much sense. But if you need those less common, those rare, difficult to detect species, the ARU is definitely the way to go. Real quick, next steps. We are going to be doing some distribution modeling, taking all of this data, plus some partner data, some secretive marsh bird survey data from CDFW and Sassoon and some other partners, and looking at those, analyzing those bird habitat relationships. Then we're going to do some scenario development. So forecasting the impacts of restoration and land cover change based from climate change, and looking at making those comparison maps. And then using that information to then update the Delta multiple benefits framework. So assessing, and then using that to assess the potential impact on multiple benefits of different restoration scenarios that might be under consideration. And with that, hopefully we are going to be helping decision makers identify the most important areas to invest in restoration, both for birds, for bird habitat quality in general, obviously, but also these other benefits. That's a whole other talk, so I won't get into all those other benefits, but carbon sequestration, social benefits, economic benefits, flood protection benefits, those types of things. So with that, I will say thank you, and thank you also CDFW and Prop 1 for the funding of this project. Feel free to contact me or Christy for any other questions. That's it.
I think we can take one question.
Yeah, Bruce, or in the back.
We've heard your question.
Oh, yeah. In the back.
All the way in the back. I was wondering what happens if you have overlapping bird calls, or how the system kind of deals with that.
Like it with overlapping bird calls in the audio from the ARUs?
Yes, exactly.
It'll still get picked up. It will get identified. And since we're primarily looking at presence absence, that's really all we need. Like I said, if you want to know the number of individuals of a particular species, the ARU, it's not there yet. The analysis might, the algorithms might get there at some point, or at least come up with some proxies like, song frequency as a proxy to abundance, for example, but that hasn't been figured out. And like how to do that for something like a marsh wren that just calls all the time and there's lots of them, that's gonna be tough. Does that answer your question?
If anyone else has questions, you can catch Julian at the break.
Thank you.
And with that, I'd just like to remind everyone, we're going on break now. Please be back at 3:45 for lightning talks. And I also just wanted to thank those that contributed to the art display upstairs. I've removed the things that were hanging from the wall. They're all laying on that desk below if you'd like to pick up your piece this afternoon or tomorrow morning. Thanks.
All right, folks, our last session of the day will be chaired for you by Mr. Brock Huntsman, and it's going to be a bit of a potpourri. So,
all right,
take it away.
All right, well, thanks everyone for coming. So this is the last section of today's talks. So this is the lightning talks. So obviously we want to try to go quick. If you have any questions, if you want to stay after, I assume we can probably sit down and discuss those, but we won't have enough time in between talks to actually go through questions. So the first talk will be by Trishelle Tanner, entitled Strategic Communication for Collective Action in Wetland Management and Restoration, Engaging with the Public, Landowners, and Agencies. And I better bring it up. Should be good.
All right, thanks for sticking around for the last session. I'm going to try and get through this in this short amount of time. I'm going to talk to you about some of what's been brought up in the plenaries from today and yesterday about how do you actually get your science out to people who use it. And before I start, I want to thank we call it Team Phragmites, and these are the social scientists on the team, and especially my postdoc Gabby, who was a de facto therapist for all of the landowners and agency folks that we asked to share their vulnerabilities about communication. So as a recovering biologist who has bridged the gap to social science in the last 5 years, I struggled a lot with thinking about how human behavior is non-rational and how we have to think critically about not just using facts to convince people to act on certain management outcomes. And what I landed on when I joined a social science nonprofit many years ago was communication framing, this idea of framing theory. And what this does is it centers cultural values and community level action based on a lot of psychological and social theories that predict and shape human decision making. And from all that theory, I can distill that into one takeaway for you, in that sense of belonging to a community can be driven by how people build literacy and how they develop personal agency to use that knowledge. So we apply this to thinking about wetland restoration and invasive species mitigation and management in two different studies that we did here in Sassoon Marsh. The first one is on the right, where we looked at public and scientist perceptions of wetland restoration and the values around wetland restoration, looking specifically at the emotional responses we can leverage to those actions to kind of collate community-derived solutions. We also worked with specific landowners in Sassoon Marsh, so the duck clubs, to think more critically about invasive species mitigation for Phragmites, to help them allocate resources effectively and work together, not just work within the boundary of their own parcel of land. And you heard a little bit about that earlier when Gabby Keeler-May talked about spatial prioritization. So this is kind of the implementation arm of that. And I'll just, since I don't have much time, jump right into the results. We evaluated trust among people to first determine who should be delivering the message of what to do. And I think it's no surprise to us that that person is none of us in this room. It is someone who shares cultural values with the people they're trying to communicate with. So for this Phragmites project with landowners, we kind of unsurprisingly discovered that they only trust one person, or one agency, the Sassoon Resource Conservation District. They don't trust any of the other agencies operating in their domain. And we kind of observed that anecdotally, but they also shared that with us in interviews. And we kind of parsed these attributes of a trusted messenger into those shared cultural values, having moderate management power, so ability to disperse resources, but not the source of those resources, and also low regulatory power. They don't like dealing with people who have regulatory power. And this is unsurprising because this agency was the product of their clubs coming together to organize for land protection. When we looked at what values were motivating action within the community at large, so this is the public and scientists, we discovered three main cultural values that kind of tie us together. I know this is a little hard to read, but qualitative data is tough to put into a presentation. And these values line up with research that the nonprofit that I worked with have put out for the last few decades. So those three values are protection, responsible management, and stewardship. And the exact framing of the phrase is really important. So we tested the words specifically in the order they appear here. So it is important to protect wetlands because they have unique habitats. It is our responsibility to be good stewards of our wetlands, and wetlands are vulnerable habitats, and it's important to manage them responsibly. So these were really important for the public and getting them to buy into whatever scientific message you were about to deliver. We did the same thing with landowners, and we kind of shamelessly tested the value statements that were being used by agencies, whether they knew they were using a value statement or not. And they contained some of these same themes. We also tested the fallacy that economics was the way to motivate people to do something. And these economics related value statements scored actually the lowest, statistically significantly lower than those three other values: protection, responsible management, and stewardship. And when people are choosing the language that they want to engage with, they're overwhelmingly choosing things that are positive in association. So people are looking for hope. They're not looking for fear or some sort of, regulatory body trying to force them to do something. I won't go into this here, but you can ask me later. We have a full set of tested metaphors for talking about wetland restoration in science. And we did this painstakingly through interviews with different stakeholders in different diverse stakeholder groups. Scientists tended to increase the complexity of the message, and the public tended to decrease it, unsurprisingly. And when we go back to our set of landowners that we were working with in Sassoon Marsh, we found again and again that they don't really care about the financial benefits. They want to feel like they're part of a community that values the same things that they do. We unexpectedly uncovered a lack of intergenerational planning, so this really jeopardizes our collective action. And we actually have an exercise for you to participate in the end here. This is the main takeaway message from these 4 years of looking at how to build wetland restoration literacy in Sassoon Marsh and the Delta. So identifying and relying on trusted messengers, shared values, and hopeful language promotes collaborative approaches or collective action. Now, if you want to know how your thinking measures up to some of the people that we interviewed as part of this study, you can take our quiz and it'll tell you how many words and frames you have in common with our duck club owners. And I have paper versions of this. If you want to come find me, I can give it to you. And if you have questions about this, go ahead and email me. Thanks.
All right, so our next talk is from Shoko Shinbrat. Lessons learned for interweaving traditional knowledge and Western science in the San Francisco Bay Delta.
Thank you. So I wanted to thank you all. That was an amazing talk. So I'll try to follow up after that. My name is Shoko Shinbrat.
I'm a scientist at the Delta Stewardship Council. This project is really a labor of love and could not be done with our amazing team at the council. So wanted to start out with acknowledging, yeah, what a labor of love this has been. Excited to show you what we've been working on. So as most of you know, agencies and interagency programs really have been grounded in this best available science. And that is historically excluded traditional knowledge. And I kind of asked myself, why is that? Well, maybe at the most basic level, it's that TK is misunderstood. So TK is kind of best defined as this combination of practice, knowledge, and ethics, which is often why it's called a way of knowing. And the word tradition might kind of make you think like old or static or dead, but tradition and traditional knowledge is really alive, experimental, and evolving. But interweaving TK and science will really require trust building, which is why I love that you brought that up. And for tribes, that will require ameliorating past harms and also using kind of more participatory methods. So thinking about, how is TK being recognized and applied in California? So in 2019, California Governor Gavin Newsom actually apologized to tribes for violence, disappropriation, and dispossession of lands. And in the spirit of healing, encouraged all state agencies to co-manage land with tribes. And 3 years later, CNRA launched its Tribal Nature-Based Solutions program. And in December, the Fed finally caught up and recommended federal agencies build relationships with tribes and apply TK. So there are a lot of participatory models. I won't go into all of them, but science kind of typically operates under this model too. So where research in that red line has the answer to the problem, and it'll deliver the answer to these tribes, communities, policymakers in blue when it's ready. So some research may, consult at the beginning, some consult at the end, some do both, but really healthy participatory practices is best exemplified by Model 5, which is this continuous engagement with tribes, communities, and policymakers to co-create this new knowledge. So what are some benefits? So the benefits of participatory science and use of TK are perhaps most obvious in things like cultural burning. CAL FIRE has this tribal wildfire resilience grant that supports Indigenous fire management while also providing funds for capacity building of the tribal workforce. And science has really already shown that low intensity fires can provide habitat for wildlife and stimulate plant growth. But because these really long-term tribal collectives are pretty rare, there are still gaps in understanding of, why what low intensity fire might help, for instance, understand understory plant diversity. So really highlighting the need to expand these partnerships. Other agencies like the U.S. Army Corps of Engineers Thule Restoration Alliance said that TK has been more useful than engineered solutions, which is quite a statement for a USACE. So they've been using funds for science-based improvements to erosion prevention and flood control, but also providing culturally important species for tribal ceremonies, sustenance, lodging, etc. So farther afield in Alaska, scientists really rely on tribes during cold winter months that are often inaccessible to do that ground truthing for things like satellite data on sea ice. So one really cool example of Indigenous monitoring in action is Siku. It's a social media app, and it really highlights traditional knowledge front and center with sightings of walruses, hunting reports, description of sea ice extent so that people can safely travel with their snowmobiles across the ice. And they pair that with scientific data on weather and tides. So recognizing the potential benefits of interweaving, staff at the council launched this multi-pronged effort
to explore TK, including this in-depth literature review of 89 articles from around the world, a review of agency websites in the Delta on what they're doing, interviews with tribes around the Delta, and then all of those are really informing a best practices guide that are developed by and for agency staff, which will be released on our new and beautiful tribal engagement website. So I'm just going to talk about one of these products, which is this best practices guide. And you should know that these are really evolving. So it's a good opportunity if we have time at the end to get feedback. So the first practice we highlight is that agency staff should really start to understand TK and the sometimes violent tribal histories, recognizing that each tribe is really distinct. To start that process of interweaving really begins with building and sustaining relationships with tribes, while respecting that tribes are really ultimately the ones who decide, who own the information and decide whether and how to share it. But agency staff can also start to make the process of interweaving official, and that can mean things like, recognizing tribes as experts in their own right. And of course, acknowledging that tribes and agencies are equal in these government to government conversations. So finally, to ensure that all parties really continue to learn from each other and seek out ways to improve and provide opportunities, we underline that there is importance for direct and reciprocal learning. And so why does TK matter? So what? Well, hopefully I've convinced you that TK is useful to the lives and livelihoods of tribes, but also for filling these local and longitudinal holes in the scientific enterprise. But beyond that, I want to underline that we are in a planet that is in deep trouble, and science has not solved the problem of climate change. So we need as many ways of knowing at the table to experiment our way into a sustainable future for more than just humans. Thank you.
All right, our next speaker is Brittany Davis. A year of case studies in the San Francisco estuary to inform conservation efforts of Delta Smelt. There's not, but look at her.
Okay, no timer. Here we go. Yeah, thanks everyone. I'm going to share a year's worth of cage deployments in 5 minutes. I'm Britt Davis. I'm from Department of Water Resources. I want to quickly acknowledge probably 30, 40 people have touched this project, and I'm just the messenger. A lot of great work here. So this study really started in 2020, 2021, where we looked back at a lot of individual cage deployment projects that happened in 2019 to answer specific questions. And a group of us thought, what a great opportunity to do a synthesis and summarize sort of what we learned. Can we take this sort of enclosure program that DWR has established, and do we think these cages are an effective tool to evaluate how fish respond in the wild? Can we use these cages to determine good fish metrics to understand what's going on in the environment and how smelt are going to respond? And then ultimately, tool-wise, can we use cages to understand how management actions, habitat, food actions, as well as experimental releases and supplementation can benefit the species? So synthesis question, and I'll share the wealth of data that we have, is does season and location of cages influence Delta smelt health, including survival, growth, and physiological condition? And so our study approach was quite holistic. We integrated a series of data sets, all from 2019. So again, I mentioned there were 9 different deployment events across 3 seasons. So first deployments ever were in the winter at Rio Vista and the Sacramento Deepwater Ship Channel. That data is actually already published by Melinda Bearwald and colleagues. But then we had deployments in summer and fall. Can smelt survive in the heat of the Delta across seasons? And then during fall as well. So you can see the different seasons and sites here. So in summer, we had the Yolo bypass deployments, again at Rio Vista. And then in fall, we had deployments in Sassoon Marsh, Yolo bypass, Rio Vista. And then across all the seasons, we had deployments or control cages at the FCCCL hatchery. I think I added another C there. So metrics across all these data sets collected included survival, growth, histological conditions. So looking at the liver and the gill, glycogen for energy and potential food limitation, thermal tolerance, and we have diets, you name it, water quality, zooplankton. I'm not going to show all the data, just some key information here. And then we did some information, sort of interviews to figure out what happened along the deployments and QC the data. So with the limited design, it came down to testing these two hypotheses, the effect of season at Rio Vista, looking winter, summer, and fall, and then the effect of location just within the fall, comparing sites at Rio Vista, Yolo bypass, and Sassoon Marsh. And I'm almost done. Here we go. So season effects. I'm going to show you all the data all at once, so stay with me left to right like a book. First plot I'm showing you is percent survival. And this is again just at Rio Vista. And what I want you to see here is that we have high survival in the winter and fall, above 90%. Summer, they don't do so well with greater mortality. Survival correlated with condition factors. So here in the middle figure, I'm showing you the proportional change in K. So that's condition factor, a metric of health and growth. What you should see here is in the winter, you have positive condition, so positive growth. And then in summer and fall, you're negative. So the fish are not growing. So the question sort of becomes in summer, fall, you know, where do they do the least bad? And then the condition factor correlates with these health metrics. So here on the far right, I'm showing you on the top a liver lesion score. On the bottom, I'm showing you glycogen depletion. So on the top, just look at the pink line, which is Rio Vista. You see that in the winter, they have pristine livers. In summer and fall, they're getting more lesions. And then glycogen, they have decent glycogen levels during the winter. And then again, in summer and fall, depleted glycogen levels. So this is an indication of possible food limitation or stress. So in terms of the effects of location, so again, this is just the sites within fall. We see similar survival across the sites. So again, read with me with the book. Rio Vista's in pink, Yolo purple, Sassoon green, and then our hatchery. So not as great as fall or as winter, but still decent survival above 80%. Again, looking at the proportional change in condition factor, so that's K here in the middle figure, similar negative growth across all sites. So obviously not what we want to be seeing, but it's expected given previous research on summer fall conditions and summer fall being sort of a bottleneck for Delta smelt. And then on the far right, here's looking just at liver lesion scores and glycogen in the fall, and we see compared to the control, high liver lesions across all sites, maybe worse in the marsh compared to Yolo and Rio Vista. So in summary, the good news is, across the estuary, we see generally high survival, decent health, but there are effects of season and location. Winter's better than summer, fall. The summer deployments, I didn't really show you the summer deployments here. Summer deployments at Rio Vista and Yolo. We had 100% mortality in the Yolo bypass in summer, so that data is not here. Summer temperatures are a big issue and smelt need to find refugia. We did see after the 4-week deployments, nutritional stress marked by the liver lesions and glycogen in summer and fall. We also found evidence of biofouling. You'll hear about a biofouling study tomorrow in Scott Meyer's talk. They potentially could have restricted feeding behavior in the cages. Maybe we can mitigate this with some management actions, we'll see. But overall, I think the synthesis study showed that first, we have good fish metrics to detect differences across habitats, and the cages can be used as a good tool to inform management and supplementation in the future. And lastly, like I said, a bunch of people helped us. Thank you, everyone.
All right, our next speaker will be Anthony Donahue, larval fish diets as indicators of food web dynamics and tidal wetland restoration.
All right, hello, I'm Anthony Donahue. My research in progress is larval fish diets as indicators of food web dynamics and tidal wetland restoration. My research is part of the Delta Stewardship Council funded microbes zooplankton project. Now, wetland restoration analysis typically focus on benthic macrofauna and adult fish, not on zooplankton or larval fish in the water column, such as my 3 species of focus, longfin smelt, Pacific herring, and prickly sculpin, nor do they focus on whether the stages of restoration provide beneficial food web resources for larval fish. My research is based on my advisor's prior study, which was focused on smelt and herring. That study did not have sufficient data to compare between the sites in the San Pablo and Sassoon Bay. We're using molecular methods because they're more accurate than traditional morphological analysis, and they're able to identify unknown food sources, such as the egg in the prickly sculpin digestive tract, pictured at the right. The prior study found longfin smelt DNA in Pacific herring and Pacific herring DNA in the digestive tract of longfin, which speculated that they're feeding on each other's eggs. I intend to confirm that in my study by extracting a sequence in the DNA of eggs in digestive tracts. Now, the food web dynamics that the microbes zooplankton project is focused on are microbes and phytoplankton, which are targeting the 16S gene. Eric Ortiz, who's speaking after me, will go further in depth on those. My research focuses on the larval fish diets, which are eating the zooplankton and various species of eggs on sediment, seagrass, and in the water column. And I will determine exactly what species of zooplankton, what eggs by dissecting the digestive tracts and then extracting and sequencing the DNA. Which will be focused on 18S and the mitochondrially encoded cytochrome oxidase subunit 1, which I'll refer to as MTCO1 after this. So my objectives are to identify the food web resources in the wetlands undergoing restoration and compare these food web dynamics between the different stages, early, intermediate, and mature, be comparing 2022 with 2023. Also figure out which zooplankton species are associated with beneficial habitat for longfin smelt, as longfin smelt has been listed as threatened since 2009 and most likely endangered in the near future, and ultimately help wetland restoration managers assess which wetlands are benefiting these native fishes. The title of wetland restoration collection sites in the map in San Pablo and Sassoon Bay. The colors represent the stage of restoration. The collection was done between February and April of 2022 and 2023. We did larval fish toes with the 500 micron net, zooplankton toes with 150 micron net in the wetland channels during the ebb tide when tide was coming out of the wetlands. And we immediately processed and stored the samples in ethyl alcohol. Now the work I'm doing in the laboratory, I'm dissecting the smelt herring and sculpin digestive tracts. I'm doing this with a small gauge needle, somewhat similar to an insect pen, actually smaller than an insect pen. Now prickly sculpin, they're typically about 3 to 7 millimeters in length. Smelt and herring usually about 4 to 12. So it's kind of similar to the size of an eyelash. That'd be the best size I could compare it to. Now I'm extracting the DNA from the larval fish with the DNA blend tissue kit. The main step in that is using an autolysis buffer to break down the tissues. Now the bulk zooplankton samples, I'm putting in the smaller subsamples, then using the eZNA soil DNA kit for the extractions. The data I have so far, as you can see in 2023 was a wet year. 22 is dry. So for all the total species that we caught, you can see in 2023, there's significantly more Pacific herring in 3 of the wetland sites. Now my 3 focal species of smelt herring and sculpin, again, a lot more herring, 5 to 12,000 in 3 of the sites. The main, I think, thing that's important in this data is that longfin smelt were collected at more wetland sites in 2023 and in higher numbers, except 2 of the red, a little bit lower amount, but not really significant. I still have a lot of work to do on my research. I need to finish my PCR library by finishing gel electrophoresis, using the PCR thermocycler to amplify the genes in the larval fish diets and the zooplankton, which also involves using predator blocking primers to reduce the amplification of the fish DNA. Otherwise, that will block out all of the DNA in the diet. And then I'll be doing high throughput sequencing to characterize the DNA in the larval fish digestive tracts, as well as the prey assemblage of the zooplankton, which will allow me to identify the indicator species in the larval fish diets and assess for differences between each restoration site. Now, my expected outcomes are to identify which zooplankton species there are in the wetlands for larval fishes, which based on Jungbluth et al., I expect there to be Urotemnocaraliae, Anthocyclopsis americanus, Anthocyclopsis robusta, as well as others, maybe Pseudoaptimus rubacei. I also need to plan on hopefully identifying which wetlands support the abundance of these species and in turn benefit the larval fish populations. I also plan to identify the habitats I plan to identify the habitats that are beneficial for the declining fish populations, especially longfin smelt, and the restoration techniques that recreate a native ecosystem in the water column for zooplankton and larval fish. I'd like to thank my funding sources, Adult Stewardship Council, COAST, ARCS, Nelson Scholarship Fund, and Conservation Biology, the Maxwell Hannaran Foundation, ICF, who use their boats to do the field collection, my advisor, Michelle Jungbluth, EOS, Tony, Jennifer, Annie, Britta, Max at ICF, Colin, the fish taxonomist at CDFW, and Stuart Siegel at the NER. Thank you so much.
All right, our next speaker will be Eric Ortiz, microorganisms as indicators of habitat condition and restoring wetlands.
Yes, thank you. Hello, my name is Eric Ortiz. I'm a graduate student at San Francisco State University, and today I'll be presenting on my thesis work titled Microorganisms as Indicators of Habitat Conditions and Restoring Wetlands. Before I begin, I would like to mention that my work is a part of a larger study, a project known as Microbes to Zooplankton Study led by Michelle Jungbluth. And thanks to the previous presentation by Anthony, which he covered some of the background information related to my work here. So thank you, Anthony, for that. So first of all, what is it that I'm trying to do? And so what I'm trying to do is determine how microorganisms from bacteria and archaea to phytoplankton and zooplankton differ between wetland sites of different restoration stages and locations. And so who is where, why are they there? And then eventually determine, hopefully, or when they're there, and then hopefully determine why they're there. So how do we do this? Well, some of you taxonomists know how difficult it is to identify these small microscopic species such as phytoplankton and zooplankton underneath the microscope. So how do we ID bacteria? And fortunate for me, there is a method known as metabarcoding. And metabarcoding is a process that allows us to identify multiple organisms within a sample. And so without going into too much detail about what this process looks like, I do want to give us a little bit of an overview so that we are all on the same page. So first off, step one involves going out and collecting our samples. And so in my case, I collected water samples at various wetlands. And after we collected our water samples, we then have to bring it back into the lab so that we can isolate the DNA. Because in our sample, we do not want other material and debris and other parts of the organism in our sample. We just want strictly the DNA. So that in step two, once we have isolated the DNA, we're then able to target a specific region within our sample. And in my case, I target the 16S rRNA region, which is used for prokaryotic organisms such as bacteria and archaea. And then in step 3, we're then able to sequence the PCR libraries using Illumina MiSeq sequencing, high throughput sequencing, which then in step 4 allows us to convert our samples to OTUs or operational taxonomical units, and matches those OTUs to reference libraries, which then allows us to identify the organisms. So then after that, we are able to conduct statistical analysis and do bioinformatics test, which is what I'll be talking about next. Well, actually, before that, I do want to give a little reminder of the 12 different wetlands that we see here. I want to mention that the wetlands are organized into 4 groups outlined by blue square boxes, in which we considered 4 different regions. I also want to mention that the regions closer to the ocean are going to be higher in salinity. So these are sites that are in the Petaluma Marsh or in the San Pablo Bay compared to the more freshwater sites that you see more east or inland. So just important to note that as we start to look at some of my results. So before I begin though, I do want to mention that I am only starting to scratch the surface of analyzing my data. So there's still lots of analysis to perform to be able to connect the microbial diversity to environmental data. But here I'm sharing my preliminary results on a non-metric multidimensional scaling analysis or NMDS plot. Using Bray-Curtis dissimilarity of the OTU assemblages in each sample. And so what that means is each point that you see here is a microbial community assemblage. And so the closer that these points are to each other, the more closely related the microbial assemblage is. So if you guys can see that kind of triangle at the very top, that microbial assembly is much different than the rest of the samples. And so first thing that we notice is that our samples to the left represent wetlands that are more saline, which is what I mentioned before, while samples to the right represent fresher wetlands. And so which leads us to believe salinity is a driver of community, which is what we expected. So another thing that we've noticed by focusing in on each wetland here is that they're not only similar in terms of salinity, but they're also similar in terms of geographic location. So the wetlands that are in the Petaluma region share somewhat similar microbial community. And as we move further east or inland, we also see the communities are relatively close in assemblage to each other, maybe a little bit more spread out in the Napa region. Again, if we move a little bit more east, in the North Sassoon region, we start to see a little bit more variability, but we're able to detect and see that they are in fact similarly related. Now, as we get to the East Sassoon region, we do start to see a little bit more spread out, where we have Briar Island, which is represented by the green kind of crosses, and then Thule Red, represented by the red triangles, are more similarly related to each other. But we do expect to see that mainly because of how similar they are in terms of their geographic location. And so take home here is that there are clear differences in the microbial communities. One important driver is likely salinity, but we do have to do further analysis to identify which environmental factors are the strongest drivers of differences of assemblages. And so what's next is we do want to take it a step further and start looking at the different wetland stages. So as I mentioned before, we have early, intermediate, and mature wetlands. So we're interested in seeing how the microbial communities differ or are similar in that reference. We also took samples not only in 2022, but in 2023. So comparing a wet and dry year to see what those microbial communities look like and how they are similar or different. We also did take samples in different filter ranges from 0.2 to 3 microns to 3 to 200 microns. We also didn't only just take 16S samples, but 18S samples. And so that's what we would like to examine next. And so thank you to everyone that participated and helped me get as far as I have. Thank you.
All right, our next speaker is Jim Orlando. He will be talking about our mosquito control pesticides negatively affecting Delta water quality.
Again, I'm Jim Orlando. I'm with the Pesticide Fate Research Group with the U.S. Geological Survey, California Water Science Center. And our group works to understand the occurrence, transport, and fate of pesticides in a range of aquatic and terrestrial ecosystems. And I'm going to talk to you for a few minutes about some mosquito control pesticides, which we've come to see as a potentially understudied source of contaminants to the Delta and perhaps affecting negatively Delta water quality. So over the past several years, during a number of studies that we've been conducting with a range of cooperators, including the Delta Regional Monitoring Program, the DWR, our groups began to detect 3 insecticides are used for mosquito control, and that's nail, dichlorobos, and deltamethrin in the Delta at concentrations might be of concern to aquatic invertebrates. And that map on the screen there shows the sites where we've detected those chemicals over the last several years, and we've had detections in water, suspended sediments, and in zooplankton. So the first chemical is a nailed, or if you subscribe to any of the email alerts that come out for vector control spraying events, the product name is Dibrom. You might see that in some of those emails. Nailed is consistently the most heavily used insecticide for vector control in the Delta. And as far as California goes, it's really only used in the counties that surround the Delta, which is interesting. It doesn't really have any agricultural uses in our region. The use does vary between wet and dry years. And if you look at the graph on the left, which is pesticide use data from the DPR's PUR database, it's heavily used, up to 120,000 pounds or so per year in some years. Take note of the asterisks above several of the years. In those years, several counties did not report their vector control use of nailed for some reason. And so those where it's looking like it's a decline in later years is mistaken. And it's either steady state or maybe even rising for that particular use. Nailed rapidly degrades to dichlorobos, which is another organophosphate insecticide. They're all highly toxic. This pesticide is typically applied by aircraft over wide areas, mainly to agricultural areas. In the Delta region and the North and South Valley. Another pesticide that's begun to be used more frequently for mosquito control in the Delta is deltamethrin. Delta Guard is the trade name. It's a pyrethroid, so it's very potent. So what I've chosen to show here, instead of pounds of pesticide applied, is the number of applications. And as you can see, those have been rising over the years. As far as we know, the reporting is accurate on those from the different counties. Typically, this product is applied by ultra low volume ground rig. This can be applied in both agricultural and in urban areas. The pesticide is very hydrophobic, as all pyrethroids, and would be expected to absorb the sediments and vegetation, etc. But we do see it in the water. Deltamethrin does have a few additional uses in urban areas, but it's really not used on agriculture in our region. Here are some results. But to start this off, there was a study done in 2011 and 2012 by Phillips et al., where they sampled following vector control spraying events in the Central Valley. And what they found following nailed applications is they found in 8 of the 9 spraying events where they used nailed, they found 100% mortality to Cereodaphnia. More nailed is applied now than was applied during this study when this took place. So here are our detections in the graph on the left, and the horizontal bars are the EPA aquatic life benchmarks for the 2 different pesticides, both for the chronic invertebrate toxicity as well as acute invertebrate toxicity. And you can see that many of our detections are above, or dichlorobos, are above both the acute and chronic invertebrate toxicity benchmark. And the few detections that we have for nail, because again, it degrades pretty rapidly to dichlorobos, are also above some of those levels of concern. Let's see. Nail is generally detected was detected 3 times, with two of those above the acute aquatic toxicity benchmark, excuse me. And moving on to delta methrin, which again is the pyrethroid. This one has an extremely low EPA aquatic life benchmark for acute toxicity. So anytime we see it, it's above that benchmark, which indicates that it's at levels of concern. And we have seen this particular pesticide in both water and suspended sediments. We detected it in delta waterways. We've also detected these in delta island drains and in delta island ponds. And again, both in water and suspended sediment. And we also detected dichlorobos in zooplankton. So just a few take-home messages. These 3 chemicals were detected at concentrations that are of concern to aquatic species in the delta. These detections followed vector control spraying events, which occurred days to weeks prior to sampling. And none of the projects that we were working on when we detected these chemicals were specifically targeting these spray events. So these are just detections of happenstance, essentially. So it's likely that the concentrations could be much higher if we sampled tracking those spray events, which is possible because spray notifications come out 1 to 2 days prior to spraying typically. And so it's possible to design a stay to specifically target those application events. These pesticides have few other uses in the Delta region. So if we do see them, we know that they're more than likely coming from these spraying events. And again, it's of course important that we control mosquitoes, vector control spraying for human health, right? But we want to understand and be able to deliver information to managers that we can, so they understand the potentially negative effects of these compounds and be able to mitigate for those potentially negative effects. So thank you very much.
All right, our next speaker will be Sylvia Angles. She'll be talking about light deck technology, evaluating a cyanotoxin detection tool.
Good, thank you. All right, so my name is Sylvia Angles. I work at the Department of Water Resources, and I'm gonna talk about water quality concerns, but taking you back to microorganisms. So I'm gonna present you a pilot study using the light deck technology to measure cyanotoxin concentrations. And this study was designed in collaboration with Daphne Gill and Ted Flynn of DWR. So in case you missed yesterday's wonderful session on phytoplankton, cyanobacteria half-algal blooms, or CHABs, have been a problem in the Delta since the past 2 decades. CHABs have been mostly formed by microcystis, but lately blooms of other potentially toxigenic cyanobacteria have been observed as well, mainly Dolichospermum anaphanizomenon. Microcystins is the most frequently detected cyanotoxin linked to microcystis blooms, but other toxins have been detected as well, mainly cylindrospermopsins and anatoxins, for example. But not all potentially cyanotoxic, cyanobacteria, sorry, can produce toxins. There are toxic strains, non-toxic strains. So we need to measure toxins in the water to fully evaluate potential risks of potential toxic events. So we have 2 technologies, two methods to measure cyanotoxins. Basically, there are 2 main standard EPA-approved laboratory methods, ELISA and LC tandem MS. But these methods are slow. It can take several days to get the results because generally they require a third-party lab. Samples are sent to these labs and they take some time, and they are also pretty expensive. So we are in the need of rapid methods. There are a few options, mainly strip tests that can be used in the field. They are rapid. We can get the results in hours. but the results can be difficult to interpret. It depends a lot on the experience of the user. And basically, it relies on the brightness of a line, like you can see in the slide. And the problems also are that they are semi-quantitative. And so we need we need new technologies. So in the recent years, the light deck technology was developed to basically obtain in a rapid assay, which takes 10 minutes to do, easy, cost effective, and we can obtain actual concentrations of microcysteines and cylindrospermopsins at the same time. However, it's not EPA approved, so it's still considered like a screening method, similar to the strip test. And results need to be confirmed with these standard lab methods. So in this pilot study, what we wanted to do is to test the light deck and see how the results were compared to the lab standard methods for microcysteines and cylindrospermopsins. Basically, we were interested in comparing the positive and the negative detections with both methods, if the light deck was reliable, and also we wanted to see how the actual concentrations compare. So for that, I collected grab samples at 7 stations with the red dots during the environmental monitoring program's monthly surveys on board of the Sentinel from May to November 2023. I collected samples from the surface and also from at 1 meter depth using the flow-through system on the boat. And then I also obtained samples of microcystis blooms in 2 locations, Cache Creek, surface samples collected in July and August 2023. And then also from Discovery Bay, from the surface and scum samples collected in September. And Discovery Bay is a hotspot microcystis bloom spot. And a little bit of methods. I also collected a subsample for cyanobacteria and phytoplankton identification in the microscope. And for the toxin analysis, I collected a sample that I sent to the third-party lab. And this lab analyzed the samples with these standard lab methods, ELISA and LC-MS, tandem MS. And then for the light deck, I collected the samples in 2 different volumes and several different volumes, which is out of the scope of this presentation. And some results of the EMP stations, we had several potentially cyanobacteria, toxic cyanobacteria. Microcystis was not the dominant. And there were no microcystine or cylindrospermopsin detections, neither with the light deck or with the ELISA, which is or the standard methods, which is good because there was agreement and there was also no toxin detections. And from the microcystis blooms, I got positive detections, and they both with both methodologies, but the light deck underestimated the results. And the higher the concentrations of toxins, the higher the underestimation. The problem was that the light deck had a narrower quantification range, which was not the expected range. So we had to dilute the samples, both the lab and me, and that might have contributed to the difference. So the main takeaways is that the light deck is a very rapid assay. Takes less than 50 minutes. What takes longer is the sample preparation, but the results seem to be consistent based on negative detections and positive detections. But there was this underestimation of concentrations by the light deck, but that needs further evaluation because I only was able to get 4 samples basically. And I would like to acknowledge EMP team and the Sentinel crew and Andrea Cast because they helped me a lot with the field work and for the good times. And also my section, mostly the people that helped me analyze the samples with the light deck and contributed getting samples. And thank you so much.
All right, our next speaker is Matthew Murphy. He'll be talking about a comparison between eDNA concentrations and loach capture in the San Luis Wildlife Refuge.
Hi, thank you all for being here. My name is Matthew Murphy, and we are going to take a look at eDNA concentrations versus loach capture. So, I'm a technician at the Lodi Fish and Wildlife Office. I've been there for almost 2 years, and I had the pleasure of working on the 2023 loach study last spring. So, some background. So, this is a confusion matrix, and it's good at doing its job. Confuses me. So the 2023 study was predicated on this 2022 pilot study, which used the confusion matrix to compare eDNA against minnow traps. So when eDNA was negative, the two methods agreed 100% of the time, but the issue lies with the false negatives right here. And basically we want all of those to be in the true positive next to it. So moving on to the 2023 loach study, we decided that by adding 3 traps per site instead of just 1, we'd increase our detections and decrease that false negative box. So the 2023 loach study was located on the San Luis National Wildlife Refuge down by Los Banos, California. So right on that red star. And eDNA samples were taken site by site to determine if loach presence or if loach were present at the site. If there were, we deployed 10 sites or 10 traps per site. If they weren't, we did not deploy any traps at that site. We moved on to the next one. So here we have eDNA collection followed by the 10 traps being set, assuming eDNA was positive, followed by a third trapping or a second eDNA effort at that same site to determine if loach were still present. Give you an idea, this is a site-by-site heat map of eDNA concentrations from our third sampling effort. And you can see there's a little bit more activity down on that southeast corner, and we'll get back to that. And this, a week later, is our catch for those sites. So you can see there is some higher catch down on that southeast corner. And this is what the habitat looked like. So waiting around in that was very fun. Lots of salve, the bank drop-offs were very deep, but this photo is representative of that habitat down in the southeast corner. So looking at these sites, we targeted sites based off of eDNA concentration. So our higher eDNA sites were the first trapped, and the results were promising as a detection method. Over half of our sites were positive for eDNA, and we got around to trapping more than half of those. And of those traps, or of those sites trapped, the majority of them had loach. So the question is, can we use eDNA to estimate abundance of loach at those sites? And the answer is not really. This figure compares CPUE against the eDNA concentrations on a site-by-site basis, and it's all over the place. Some of our highest loach catch had low eDNA and vice versa. You can see that one all the way at the top there. And this result was not unexpected. eDNA is finicky, and it relies on a series of favorable abiotic and biotic factors to really shine through. So there are a few reasons why I speculate this could be the case. So the efficacy of our minnow traps is a big one. So we know they catch loach because 88% of them deployed at sites caught loach, but the issue is a time delay. We had to send our eDNA samples back to Kramer up in Sacramento, and then we set our traps the next week. So there was a 7-day delay before our traps were set and the eDNA detections were made. The other issue is the mesh size selects for larger loach. So the vast majority of our loach caught were a around 150 millimeters, but larger loach. And we caught 2 or 3 that were about 60, and they were just too big to get through the mesh. So it's very possible that eDNA is detecting loach that are there, but couldn't be captured by us because of our equipment. The other issue could be hydrology. So the hydrology of the wetland could be a compounding factor. The wetlands flow is determined by recirculating pumps. So you're not getting a lot of new water. There could be eDNA being carried through those pumps and redistributed at the other end of the refuge. The other issue is that the variable flow level in the canals, there are a ton of sluice gates that affect water height and flow direction. So when you're setting a trap, like this one right here that's out of the water, we set that one in close to 2 feet of water with the flow going the opposite direction. And we pulled the traps 12 hours after deployment. So in that time, the water was going the other direction and our traps were out of the water. This happened a few times. So it's difficult to determine if loach were present at that site because we weren't fishing. All right, eDNA collection and loach biology. So loach are nocturnal and we're not. So we were sampling during the day, and this could very well lead to a mismatch in peak available eDNA in the water column. And when we sampled, we could have missed that. The other thing is to loach burrow. So when we were at the refuge, they were just planning on doing a major waterway construction. So they were draining a large portion of their canals. So by the end of our project, a lot of our sites were dry, and loach queue into that, and they will dig into the substrate. That's how they were found, was with diggers in dry canals. So here we have an example of a loach somewhat successfully burrowing, especially in that bottom one. But they don't make themselves easy targets. So yeah, I'd like to thank Katie Karpenko from Kramer for use of her ARC maps, and the Lodi office, Genadax, and the refuge staff. Yeah, thank you.
Today, Dan Ellis, IEP fish survey is running into, and does it matter?
Hi, I'm Dan with IEP Synthesis. The answer is yes, it does matter. You can go home now. So here's the problem we're facing. The IEP surveys were designed to be sampling pelagic habitats, but as you probably know, submersed and floating varieties of vegetation are expanding around the Delta. That's mediated by climate change, as well as ongoing species invasions as recently as a couple of years ago. But the literature is pretty clear that for both fish and invertebrates, for both abundance and composition, we know that if we sample a habitat that is vegetated, we will capture different biota. But our IEP surveys aren't taking account of this. So, or I should clarify, they're not taking account of this in a consistent way. So for me as a synthesis person, and for you as a data user, that's a problem. So some groups are commenting when they find veg, but as you can imagine, having been in the field, that's very inconsistent. One group does quantify veg that enters their net. Another doesn't clarify whether it's debris or vegetation, but if it's hitting the flow meter, they will note that. But there's nothing consistent across surveys. So the goal here is to work with IEP surveys to determine if they are being impacted by vegetation, kind of just try to get a preliminary sense for how big of an issue this might be. If there's not an issue, that's great, awesome. We can move on. But if it is an issue, maybe we need to figure out a way to proceed and account for this data. So I have to thank all of IEP. I believe I got survey responses from every different IEP survey. The data that I'll be showing you come from 600 different samples. And I wanted to say something else about this. All the study questions were designed by myself, but in coordination with the field leads for these projects. The idea was that you could add this to your sampling protocol and do it in less than a minute with very, very little extra effort. So I'm just going to really quick jump into the questions and what they are intended to help represent. So first off, did you have to move your sample due to vegetation, or did you have to cancel it? So this could be your beach seine site doesn't exist, or this could be that you meant to go sample over here. You had this fringing Egeria bed and you moved your sample 30 meters to the right. So that would be that question there. The next one is one that reflects the potential for vegetation to affect your sample, even if it doesn't make its way into the net. So one thing that I heard when I was initially digging into this was, you know, we know when veg hits our net, but as you can imagine, you have different predatory fishes lurking around the fringes of these vegetative beds. And that you can imagine that that might be the same for different biota. So we asked the question, did you find veg in the net's path or near the net's path? The next two questions were kind of looking at the flow meters. So these flow meters are really critical for our estimates of CPUE. And I didn't just want to know if flow meters were being blocked by vegetation. There is the potential for flow meters to be used in scenarios when they're really not quite appropriate. So the general oceanics flow meters don't really perform well below 1 meter per second. And so I did want to clarify if the CPUE that we saw was potentially incorrect or off because of that and not because of vegetation. And then the plain and simple question, did veg enter your net? And then finally, maybe trying to get at which veg is causing the problem. We looked at, and I want to note here, it's if one particular species made up the majority of the sample. So if you had 3 equally spread species, the answer would have been no for this question. So jumping into our responses, this is an aggregate of the different surveys. So there is a large bias here for the different gear types and sampling months, but I want to say that the data came from 2022 and 2023, and I believe we had 15 months of data. There's no month that was skipped. So again, 600 samples are represented here. For sample moved or altered, somewhere between 2 and 29%. Obviously, like I said, affected by the survey type of our samples are straight up not happening or not being done the way we intended them to do. We are encountering vegetation in the net's path or adjacent to the net's path somewhere between 9 and 36% of the time. Again, this is unaccounted for. Veg is entering the net. This one just absolutely mortified me. 20 to 60%. That's a pretty high number for something that we know is scientifically proven to affect the data that we're collecting. I was actually relieved to see that the use of flow meters in environments where they're maybe a little too slow for them was fairly minimal, somewhere in the range of 0 to 5% of samples. Flow meter was physically obstructing the flow meters, again, a very variable percent of the time. Submerged vegetation caused more of a problem than floating varieties, but both were an issue. And this is my summary slide. I'm actually just going to stick it here and let you just take this in and imagine what that does to your data for a second. Imagine you're trying to understand long-term trends in abundance because you care about a species that's near extinct, and you recognize that you're sampling a different habitat through time, or you want to understand how restoration is impacting the environment, and you compare it to data from decades ago, again, but you're representing a slightly different habitat type. So I would argue that we should be digging a little bit deeper into this issue in a more rigorous way, hopefully with a more even sampling distribution across gear types. And I also want to invite you to get involved in this if this is something that either concerns you or something that you find yourself very excited by. So thank you.
So this actually concludes our lightning session today. I'd like to thank all the speakers. being able to present a 7-minute talk and concisely is very difficult. So thank you for doing that. We're actually like 3 minutes ahead of schedule, so that's great. And slide, go ahead.
Yes, thank you very much to the lightning talk presenters and all of the wonderful presenters today, the poster presenters. It's been an awesome day. Tomorrow is going to be another awesome day. So if today is sadly your last day in person, I know you'll be joining us remotely tomorrow. Please drop your name badge in the bin on your way out. If you'll be here tomorrow, keep your name badge, obviously. And if you are a poster presenter, please be sure and pack your poster up by about 2 o'clock tomorrow. If you want to take it home tonight, feel free. We're not going to have an open poster session, but the poster hall will be available until 2 o'clock tomorrow if you missed anything. And if you have talks for tomorrow that you haven't uploaded, please do so today or first thing tomorrow morning. We'll see you tomorrow. Have a lovely evening.